to the heights
more than 40 students
of
are
your goals.
there.
In a conflict
So we
between
have
the
a relief
heart
work
and
due
the
to the
brain,
years.
follow your heart. Talk to yourself at least once in a day. Otherwise, you may miss a meeting with an excellent person.
So in the time
in
of
this
years
world.
and then cyclone and then COVID-19,
Who is
in our
helping you?
small barrier, we
Don't
try
forget
to
them. Who is loving you? Don't hate them. Who is believing you? Don't cheat them. Anything that makes you weak,
this is the dry ration we provided to them.
intellectually and spiritually, reject as poison. A man is not poor without a rupees, but a man is really poor without a dream and ambition. Fill the brain with high thoughts and highest ideals. Place them day and night before you, and out of that will come a great work. Take up one idea, make that one idea your life. Think of it, dream of it, live on that idea. Let the brain, muscles,
Not only
nerves,
that,
every
we
part
have
of your
some-
body be full of that idea, and just leave every other idea alone. This is the way to success.
When the flood was there in the West Bengal, so we have this relief work. Not only that, we
Don't
have winter
overthink.
relief also.
Overthinking
So we
leads
distributed
to negative thoughts.
the clothes to the older people as well as
We
Thank you, Ankita. Good morning to all. When they start, first of all, I thank all the organizers, Professor Bisham, Professor Misra, and the entire BioHill team for inviting me here and share our research experiences with you. Past 2 days was active, packed, jam-packed, but I see that everybody is seriously listening to the talks and very interactive. After presentations, there are a lot of questions from the audience. It's very happy to see those things. That means you have reached, what you want to tell to others. I am from Sri Chitra Tirunal Institute for Medical Sciences and Technology. Yep. Hi. So I think I should finish in 10 minutes, probably. There are more slides, but I will try to squeeze in without missing the important points. I don't try to explain much on tissue engineering and all those things, but I will touch upon all those, why we want to do this kind of work. We started this bioengineering program in 2016, the 3D bioprinting program, with a vision proposal by Am I audible? Yeah. So vision proposal by Professor M.S. Valyathan is going on. So how can we engineer such a complex issue? The cells are very varied. It has got multitude of functions. It's not easy to manipulate or bring all those things in one place and in the lab, outside the body. It's very, very tough. Next, please. So all these function, if we say, when these functions are lost, we are left with only one final therapeutic approach, that you transplant. And the donor tissue is very scarce. We know that the supply to demand is very, very large. So if we say this is the demand, but this is the supply, very less. And to show the magnitude, if we have all these waiting list people, if you ask them to board flights, it will fill up 586 Airbuses. So that much people are waiting for organs, different organs. Sorry. So how this concept of transplantation and bioengineering of tissue come in? I show this slide everywhere, repeatedly I show this. I like this slide very much. Suppose this truck is very rugged truck. So there is nothing there, it cannot be run, there is no windshield, it's worn out truck. But if you go to the service station and then ask the engineer to replace back, he will buy things and he will make it in a proper condition. Is that possible? By purchasing organs. That is what the vision concept. Now, we are all working on tissue engineering towards this. There are various challenges, limitations, different things are there. We know everybody has their own limitations and difficulties in achieving transplantable organs. So this is the concept. Now there comes the tissue. I don't spend time here because we are all working on tissue engineering. It is basically making biological substitute from scaffold, cells and its nutrients and then transplant it back to the patient. If we come to liver tissue engineering, there are 4 main approaches: taking xenogenic cells, putting it into bioartificial liver, or the cell is infused, or you make a tissue engineered construct and implant. Is this possible? Yes, it is possible. This is only the product from Wake Forest Institute. They have made this much things and clinically used. For example, the bladder which is shown here, this bladder was implanted in a boy, 5 years old. He is now 28 years old. So it grew with him. So it is possible. Only thing is we have to have vision projects, vision approaches, and it is not like we are following experiments, methods. We just Google and do some experiments. We should have processes which is different from what is being said now. Now, in this work, what we are going to do, we will try to address is, suppose we have a liver damage in a patient, and then say we have a biological substitute which can be implanted or transplanted into that patient. Now, we have cultured those constructs in a very nutritive, nourished environment and in the lab, highly accelerated system, and then we are putting it into a diseased liver where it has to survive and meet the function of the normal liver. Now, during that time itself, the transplanted liver will experience a lot of helps in priming the liver to regenerate. That means the tissue engineering method. Now, normal tissue engineering, the liver is a solid bulk organ, so you cannot have a small, it is not like we print it in a small petri dish or multi-well and say that we have made liver construct. It's not. To transplant, you need bulk. It's a solid organ. Sometimes the technology that we see here might not be sufficient also. We are working on that also. Now, there we need 3D bioprinting. It's a layer by layer technology. We have cells and its growth factors and the extracellular matrix put in layer by layer fashion so that all the construct has cells inside. Normal conventional tissue engineering, we make the scaffold and seed cells. If the construct is large, the cell will not go into the construct. So there, 3D bioprinting is very handy and very promising. So we consider this program in 3 different levels: pre-printing, printing and post-printing. In the pre-printing, we should identify the cells, so from where the cells will come. That's a challenge, even now. And then the bio-ink to print. If this printing means you need ink, so the ink should be a biocompatible material. It should be a biomaterial. Should go through all the stringent tests that we do for normal biomaterial. And then a 3D bioprinter which is qualified to print and the product can be transplanted into the patient. So finally, it will be put in a bioreactor and so this is the process. In the pre-processing stage, we will image and then make a CAD model, and then this will be mixed with the cells and the biomaterial, and the growth factor will be mixed and loaded into the printer, and finally it will print. So that is our printed construct. So we need bio-ink. The bio-ink itself is important part of bioprinting program. We cannot take any material and just print. There are a lot of science behind it, technology behind it. Ink which can print liver cells. Practically, I will tell you this. If you isolate hepatocyte, put it in a centrifuge tube, you shake it, it is useless. Because it will lose all this function, when you stir it or shake it. So much sensitive cell. From where we get cells, and we are thinking about 3D bioprinting, we need millions of cells. We culture in this much stacked large T75 flasks. So we need a cell factory to generate that much number of cells to print liver. We started it into GELMA, gelatin methacrylamide, which is all well known now. And then we characterized that. And we checked the cross-linking ability, so it will be sponge-like, it can be stored in room temperature, and then you put it in culture medium, it will dissolve. You irradiate with UV, it cross-links, and then it is printable. So printability means, in simple terms, it's a big concept, but in simple terms, you each should print the design that you give. And we should print clinically relevant size. It's not small, you need large constructs. Yeah, next. So here what we have done is we have mixed with FG2 with GELMA, bioprinted, crosslink with UV, and then construct was maintained in culture medium. So in this process, can you please click there on the video? Yep. So basically in this, it prints in different multiple places and all. Now we are talking about liver, so we should have liver functions there. Albumin, urea, cytochrome P450 activity, LDH release, everything. Next. So minimum 7 days we tried. We have gone up to 28 days. Next, please. So now, how we organize these cells into liver? Now, if you take a bioink, put cells there, and then print, it will be single cells coming as filaments. That is nowhere related to the structure what we are talking about. So we are trying to make spheroids from these cells. You can see the spheroids here. We have designed a chamber which can be reused, which will give at least like 10,000 spheres in 3 days. And then we have optimized all these parameters of printing, and then we evaluate the function, all these functions of the liver. Now we have 2 products from this project. One is the bioink itself, and the second is the test system. The test system is now open to the customers. We have a lot of customers coming from research institutions, so they take our facility to test their drugs and chemicals. Next. So this is the complex structure. Now how can ever imitate or recreate such a complex cellular arrangement in our printed construct? So far we have, next, click, next. Yeah, next. This will be the normal liver in histology. Next. This will be the 3D bioprinted conventional extrusion method. Next. So this is the thing that we have made, that partially we say histomimic construct, that is our concept that we put forward, that once you print it, it should, histologically, it should match the original organ. Next. Why, I mean, just before. If you see this, I always say this, see, the birds fly, aeroplane also fly, but the aeroplane never flutter its wings. That means it's the function that matters, not how it works. This is a histological architecture which shows all the cells and bile canaliculi structures. And we have checked all the cells of the liver in there. So we have another set of data showing where these cells are, because immunohistochemically, whether it is matching the normal liver or not. Now this about coming to this thing. Now we have a liver construct in hand which can be implanted in rat liver, which already been done, normal thing. But we anticipate that the transplanted liver construct will experience lot of stress from the patient, right? He is taking medicine. He cannot move. It is nutrient depletion. And this is a live construct which is taken from the lab and putting it in a, I think next, done. This is what we have to tell. This is a concept that we are putting forward, that the construct itself has to be capable enough to take the stress and this, all the adverse environment that you put it in a host. It's a small team. I thank everybody, funding agency, our directors, and yeah, these are all their work. So I thank them all. Thank you very much.
If anyone having any questions, please.
Printed construct. Now spheroids is 200 micron radius, 500 micron dia. These are organized liver constructs, micro tissues.
So they were independent.
Independent spheroids. Then that is printed.
That is printed. So I mean, so I'm trying to understand what would be the logic behind independent such sort of construct in terms of creating the liver tissue. I mean, one can create such sort of thing through pipette also, using say 20 microliter deposition of say some gel in a well plate. So if such a creation would happen, then what would be the relevance of
so
it?
This chamber which I showed, if manually the student will do, it takes 20 minutes to pipette that. If you use an inkjet by a printer, it will do in 40 seconds. So it is high throughput.
So it's high throughput.
The dimension of cell requirement that we talk about in 3D bioprinting is enormous. The dimension has changed. The dimension means dimension in every aspect. Number of cells, the functionality, the size of the construct. It's not a simple kind. We can print a small nail size thing inside a plate. That's not what we require. So from where such kind of cells will come? Now if we have that cells there and printed, it takes so many days to get into a liver construct. It has to mature into. We have to cut down the in vitro time considerably, maybe like 2 days, 3 days, and make it ready to be transplanted. That is where that was.
One more question, sir. So if you have tried with primary hepatocytes as well for this printing process?
Yes.
So what was the
current
source?
project is to isolate cells.
From source?
Right.
Right. Okay.
So we are now getting ethical approval for human liver also.
Okay.
And so this rat, because we are working in primary liver cells for 20 years now. So we have a standardized protocol, we have all this set up to get 95% viable functional hepatocytes from liver. Okay. We have also decellularization, whole organ decellularization, which is very tough. Suppose we get a large human liver now, we can decellularize in 3 days. So we have all this set up there now. So, what I meant to say is we take primary cells, print it, transplant back to a rat.
So I'm trying to learn from your experience. Like if I print, say, in a linear fashion, not in, say, a spheroid, say, linear integrated fashion, say, some kind of a square shape, then what would be the difference between independent, say, individual spheroid with compared to, say, a square form of linear pattern? So
there are two things. You can inject the spheroid itself. That's enough. But the problem is it will clog. Because normally it will be injected into the portal vein. But as and when it reaches the branches, it will clog there. But it will remain there. There are many studies. Our clinician also, all our project will have a clinician in the group.
Sure.
That, it will reach there, but it will be like clogged in the first branch,
Okay.
So you remove a portion and put a construct there. That is practical.
Okay. Thank you, sir.
There is another method of getting scaffolds. Like if you infuse pronase, the whole hepatocytes go.
Right, sir?
So this is little better because the other parts of the liver, which is around scaffold, remains. So you could convert a diseased liver into an active liver through this.
Yes, sir, absolutely right. There are two ways to bioengineer tissues and organs. One is, like sir said, decellularize that and recellularize because it is structurally similar. So there are also work going on. So one PhD student is working on decellularized liver matrix.
Thank you, sir.
Thank you very much. Yeah. Thank you.
Thank you, sir, for the informative talk. The next speaker is Dr. Mahesh Kumar Saha. Dr. Saha pursued his PhD in biotechnology and medical engineering at NIT Roorkee. He was a faculty in charge at Innovation and Entrepreneurship Club, NIT AP, and in 2017 to 2018. He was a member of International R&D Committee, MIET, Bhubaneswar, Odisha, and is currently an assistant professor at Department of Biotechnology at Dr. B.R. Ambedkar National Institute of Technology, Jalandhar. He was honored with outstanding research in biomaterials and tissue engineering in 2020 by Venus International Foundation, India 2021. And he was also awarded National Eminent Young Researcher in 2020 by International Institute of Organized Research India. He was also awarded Bharat Vikas Award in 2017 for contribution in the field of tissue engineering from the Institute of Self-Reliance India, Bhubaneswar. His research interests includes biomaterials for tissue engineering, stem cell research, nanotechnology, biopolymer for environment engineering, waste valorization, computational biology, and drug delivery system. We welcome you, sir.
Hello? Fine, thank you. Good morning, everyone. So, thank you for the nice introduction. So, just it is always a concern that how the natural waste that is being wasted and that is adding to the ecosystems can be best way utilized. So, that way we have come for the tissue engineering applications and the tissue engineering application of the avian eggshell membrane. So, it is always the questions that is still unsolved, that who has come first, the hen or the egg. That is still unsolved by the ChatGPT also. But one thing is very much clear, that from the second part, that is the egg, the different components of the egg can be best valorized for the tissue engineering applications. If we go to the different components of the egg, here you can see that already egg albumin has been utilized for the tissue engineering applications. Some groups have already attempted with the egg yolk also. But the eggshell membrane part is very much underrated in research, but it is a very much resource of the varieties of natural components that can be best suited for the tissue engineering applications, particularly the bone tissue engineering. Why? Because we are telling that this is underrated, because there is a very limitations of using it, that is the processability of the eggshell membrane. So from the Japan and the Taiwan, even the scientists are here. So they might be knowing that how much it has been directly utilized for the wound healing process without any processing it. But if we are able to somehow utilize it in such a way that the processability of this material can be enhanced, so obviously we can use it far better way for the tissue engineering applications. So this this eggshell membrane, if we talk about, that is actually the globular varieties of proteins are there, that is actually strongly bonded with the collagen proteins. And since the globular proteins, that when we are isolating it, that can have the, when it is in the natural form ESM, that is has a rich source for the arginine, glycine, aspartic, RGD sequences. And all of you might be knowing that how much it is important for the improving the biocompatibility. Besides this, it is also containing varieties of proteins, osteocalcin, osteoceallin. So all these proteins are very much helpful for the bone tissue regeneration. Next, so one more, we have attempted initially with the silk fibrin just to improve its property, because we just wanted to check that how much efficient it is to improve its property. And we found that when it was integrated with the silk fibrin, it was able to not only culture over that, but it was able to influence its physical properties as well. Next. And further, when we were satisfied with its biocompatibility, we further went with the in vivo studies also, and we found that it is not showing any immunorejections or any kind of chemicals or toxins that it is producing when it is inserted in the in vivo studies. Next. So obviously the ACP part many groups have already done, but what they are ignoring actually the collagen part. When the different components that is already integrated in the eggshell membrane is also related to the bone tissue engineering applications. But what we can do, if we are isolating it, we are extracting those components from the egg cell membrane, what is left behind, that is the pure collagen. So if somehow we are able to extract out pure collagen, obviously it can be migrated to the variety of tissue engineering applications, and it won't be limited to the bone tissue engineering applications. Next. So here we have tried to extract it. So when we optimize the process, we can see the optimal temperature was there. And what exactly we do? We firstly go for the enzymatic treatment first. And before that, what we do? We separate out the ACP part. ACP part is the globular part that is bonded with the membrane. So once ACP is removed, behind is the collagenous part. But that collagenous also we can't use it. What we have to do? We have to partially digest it. So for digestion also we have to optimize the process. And we found that if we are using the enzyme pepsid, at 350 unit of grams, it will be maximum way it is giving the yield. Likewise, if temperature we talk, so temperature and pH is also there. So that way it is giving pH of around 3. So at that time the maximum yield is there. Next. Likewise, sidewise we were also checking that what the physical property it is changing with the different process. Next. Likewise, we also saw the hydrophilicity of that material that we have digested, extracted collagen, have what extent of hydrophilicity that further it can be used for the skin tissue engineering or not. So that we found that it is very much hydrophilic and that it can be further used. Next. Next. Okay, so likewise the process was optimized for the extractions so that the digested collagen can get. Then our next purpose is that how we can modify it, how we can modulate it in such a way that it can be used for some kind of skin tissue engineering. Next. So for that purpose, we have to go for the hydrogel preparations. In that also, we selected a very simple method for the sol gel transition. And when it went through the 6 cycles beyond, obviously in 5th cycle, it just started making the gel. After 6th cycle, it was having some mechanical strength. So once we are getting the mechanical strength, then only further we can go for further studies. Next. So likewise, it was very much porous, and likewise, we can see that it is having some porosity, and those porosity actually we have to match for the skin tissue engineering. Next. So obviously, before proceeding, we have to go for the biodegradability test. We have to go for the what are the different functionals exposed, and that will be exposed to the cells in future when we will go for the in vitro or in vivo. So those characterizations will further confirm that okay, it is suitable for the skin tissue engineering. Next. Or before that, just one slide. Okay. So initial preliminary in vitro study with the MTT assay that was also indicating that when we are getting the gels, at that time we have the different concentration that we had prepared with the digested collagen for the 0.5, 1, 1.5 and 2. Out of this, it was better for the 1.5%, and that could have been, we could have further processed. Next. So further when we started making with the different compositions, so obviously likewise if digested collagen is there, it is again we have to add something so that we can make the hydrogel out of it, because without that it is very much brittle. So to make it more compatible, more with the mechanical strength, we are preparing and mixing with the PVA in the different ratios. So we prepared for the 10-90 ratio. Here 10 is a collagen part and 90 is the PVL. So likewise the different ratios we are prepared out of that. Next. Okay. So initially we also proceeded with the antimicrobial study, and we found that it is not causing any inflammation or inviting the microbes to the material. So it was completely antimicrobial property that it was showing. So we had also planned to utilize this antimicrobial property of this material for some other applications as well. But anyhow, for this tissue engineering, skin tissue engineering also, we require that the material should be antimicrobial. Next. Okay, so one part was the egg cell membrane where the ACP part was there, and another was the collagen part. Now what is remaining? That is the egg cell cuticle. So that cuticle is basically composed of the calcium carbonate, and it can be a very rich source of the calcium ion. So likewise, whenever we go for the tissue engineering applications, either we go for the direct calcium phosphate resources, or we go for the calcium ion resources. So here what we have the calcium ion resources. And further, based on that, whether it is the calcium phosphate resource or calcium ion resource, accordingly, the different methods to get the calcium phosphates are here. Further, we have to go for the thermal calcination or alkaline hydrolysis or subcritical water or combination methods of all this, so that we can, from the bones or fish scales, we can make the biomaterials out of it. So likewise, if the calcium rich sources are there, a very common now in trend is the hydrothermal. So that way we can get the calcium phosphate. And here what we made that already we have the calcium rich source from the calcium eggshell cuticle. But obviously calcium, it has to be transformed to the calcium phosphate. And initially whatever the methods were there, it was actually giving the hydroxyapatite. None was the reporting for the, or efforting for the beta TCP. Anyhow, they tried, many they tried, but they were getting the biphasic material, not pure beta TCP from the eggshells. So just what we did, we just included one additional method in between. That was the ball milling method. So when we introduced the ball milling method, through this, when we prepared with this method, so we were getting pure beta TCP. Next. Out of those pure beta TCP, we had prepared the scaffolds, and when the cell culture was done out of it, we were getting very smooth culture over it. And with the XRD data, okay, XRD data is not shown here. With the XRD data, it was very much clear that it is giving very stable beta TCP and very pure beta TCP. That is compatible and comparable with the commercially available beta TCP. So in labs also, you can always try, because in market you can get it very costly. So that we are only preparing for our labs, and we are applying for the different applications. Next. Okay, so here the cell culture was also indicated. So here we can say that the CPM, here it is the ball milled product one. So after 14 days, the confluency of mesenchymal stem cells were there or seen over the scaffolds. Next. Okay. So, no, just before that. Okay, next. Okay. So, just what we have to do, we have to go to diverse the different applications through the different material processes out of all this. So, since we have already prepared the hydroxyapatite out of the eggshell, talking about only the hydroxyapatite. So, we have prepared along with the PVA so that we can make the hydrogel form, so that it can make into the injectable form. Next. Now comes that if we want to go for the wound healing, so obviously we have to extract those part of the eggshell that is very much close in composition with the skin, and that is the collagen part only. Obviously we have to remove the ACP part. It can induce some mechanism related to calcium ion during the wound healing that is assisted with the help of ACP. That ACP is also the part of the axial membrane. But for the formulations, the concentration of those ACP should be very much limited. And how we can control or monitor it, obviously we have to separate it, and additionally we can add it. Before that, obviously we have no control. But base material is the collagen only. So this way, when we are putting the collagen over it, so it is able to make the papillary present in the axial membrane, that is contributing for the skin wound healing. Next. Okay, so in this future direction, only what we are presently working on, that we are trying to find some green methods using some ionic solvents, so that we can make the collagen in a very much easier way, without any contamination or the toxicity with the different compositions, so that it can be utilized for the osteocarcinoma therapy. Next. Likewise, we have, in a single step, we have developed a protocol so that we can get the chitogen and hydroxyapatite from the crepe cells. Next. Likewise, we have also developed the nail caps for the recovery of the perinecia, that very frequently the people with the vitamin deficiency of vitamin they face. That for that sake, we have prepared the nail caps, and it is very much antimicrobial, that already we have proved. Next. Likewise, I think day before yesterday, one call was also there when they were asking about that how to you work with the desalination system. So this desalination system that we have developed, in that it can be utilized for the seawater. Now you may be asking that what is the need of the biomaterial scientist here. So now we are making a biofilters that is also from the human hair only. And we are going to put at that blue pipe is there, so that is the outlet. So there we are going to utilize that biofilter that we are developing. Next. And this is the automated dialysis system. All of you know whoever are working with the proteins in the labs, they frequently go for the dialysis. And they use the dialysis membrane or cassettes. So likewise, frequently you have to change the buffers and you have to change the water with due course of time. But in this system that what we have developed, one side we have to just put the buffer or the water, and second tank is just to collect it whenever the time is set. And it is completely based on the detection of the TDS system. So since we frequently required it, so we developed these systems. And this has also awarded first prize during recent last year one event was there in IIT-BHU. So first prize it got. Next. So this is my group from NIT Jalandhar. So I have done nothing, means I have just guiding. So these are the guys who are actually contributing to all these works. Okay, thank you.
Thank you, sir, for such an informative presentation. I have a very simple question. You mentioned in your slide that it will sow antimicrobial against E. coli. I'm just curious to know, will it sow antimicrobial activity against any gram-positive bacteria?
So it is always a matter of research only when we go for the varieties of most common microbes. So out of that, we have selected E. coli and it is showing that.
Okay, sir. Okay, sir. Okay. Thank you.
Thank you, sir.
Okay.
Thank you, sir, for the informative talk. Next, we have Dr. Suman Jha. Dr. Jha received his PhD in 2009 from the Department of Physical Chemistry, Technical University of Dortmund, Germany. He did his postdoc training from the Department of Molecular and Cellular Biology, University of Connecticut. He is currently working as an assistant professor at Department of Life Sciences, NIT Roorkee. He has been awarded with Inspire Faculty Award from the Department of Science and Technology, Government of India from 2013 to 2018. Dr. Jha's research interest lies in the field of protein folding and dynamics and nanoscale biophysics. We welcome you, sir.
Forward, backward. Yeah. Thank you for the brief introduction. Myself, Suman Jha, and good morning, all of you. And I welcome you for my presentation, which is on the optimizing the nano bio interface for different applications, which we are working in our lab. So before going to detail, I just want to briefly introduce what is the amyloidosis problem, or what is basically the protein folding problem. Generally, it is associated with the old age disease. What happens in our system, we have evolved for 2.5 billion years, the proteostasis network where protein is being folded, right? Being translated, then nicely folded with the help of chaperones. And if it is misfolded, there is a system we call remodeling system, which disaggregate it, then refold it into the physiologically active form. If it can't be done, then it is being cleaved by proteasomal degradation, right? So what happens, sometimes this process, misfolded is very slow. They accumulate with time. Their population, their fraction population is not too much. So what happened, this is in vitro study, what happens, they accumulate with time, which is very lag phase, and it can't be probed, it can't be diagnosed in early stages. Then it starts in explosion phase, where they form some fibrils, and over here, these protofibrils are very cytotoxic. And there, the degeneration of the cells or tissue starts. And then in the plateau phase, when everything is done, which is non-cytotoxic, its symptoms start appearing. And so that is in vitro, happen within 20 to 24 hours. That is the IPP, isolate amyloid polypeptide fibrillation or amyloidosis, which is considered as a kind of causative agent for type 2 diabetes, 90% of type 2 diabetes. Right? And if there is certain change in condition, that lag phase is reduced to only few hours, few minutes, in fact. And that happens within minutes, within hours. They enter into the mature phase, and everything is done. And since this is so fast, and the free energy content, the thermodynamics of this aggregation is so little, so negative, that it can't be reversed, right? So it's a big headache, and since it takes too much time, so it is generally associated with the old age disorder, right? Now, why I am introducing this one, before going to my current research, so we have found, like especially me, while I was doing the postdoc, so that protein aggregation, right, the amyloidosis problem, right, so that was kind of very minute change in condition can result in this amyloidosis problem. For example, IPP, it is generally stored along with the insulin in secretory granules where pH is around 5. And when at pH 5, you can see the lag phase. That lag phase is almost around 8 hours. And as soon as it is secreted into the extracellular matrix where the pH is around 7.3, it reduced, the lag phase reduced to the 7 hour. If it, reduced to 1 hour. If it is not clear within the time period, that it may cause the aggregation and may lead to the reduction of pancreatic beta cell, that is the tissue over there, and so type 2 diabetes onset starts, right? And this is about the semen-induced, semen-mediated enhancement of viral infection, another papillomavirus protein, which forms the amyloid and brings the pore into the tissue, into the cell, which enhance the viral infection in the genital, right? So we have seen that peptide fibrillation also reduced by manifold, the lag phase, manifold, when there is some copper or zinc ion, right? And naturally, when in the semen there is very high amount of these metal ions present, which is there to inhibit such kind of amyloidosis process, right? So why especially I'm interested, because nowadays you must have seen there are many applications of metal nanoparticle, especially the photocatalytic nanoparticle, like there are during the COVID cases, COVID situation, there was certain soap which was coming with the silver nanoparticle inside it, the black, I don't know whether it was Godrej or what. So they were saying that it is having very good antimicrobial activity. That's the right thing. It can reduce your microbial load by 99%. But what after that, right? Because nanoparticle, if you use it, you can't stop it over there. They can diffuse through the tissue to your, if unfortunately diffuse to the bloodstream, it can go anywhere, right? Doesn't matter whether it's in picomolar or watt molar it is, it can diffuse, right? Obviously, for age-related disease, it won't reflect immediately. So whenever it is coming out as a symptom, you won't be able to know whether it is because of the nanoparticle or what is the factors, right? So why photocatalytic nanoparticle? Because they can promote the interaction between the interface can result in conformational rearrangement. That conformational rearrangement is two-way sword, right? It can better the fibrillation, it can better the functional conformation of the protein, or it can deteriorate the conformation that is prone to the amyloid formation. Or these photocatalytic nanoparticle can also bring the change, covalent changes, like there was certain, like they were making certain copper shield or copper nanoparticles which was having the catalytic property which could cleave the peptides, right? So that is also photocatalytic property of the copper. What if that nanoparticle can also cleave your functional protein? They can also moderate or enhance the H product formation. That is not good with the amyloidosis protein. But there is not always a problem with the nanoparticle. If we can moderate the interface, we call it corona. When it enters into the biological system, we call it corona, hard corona or soft corona. Hard corona is the metal part, and soft corona around it, which is used to cap them in the nanoparticle size, right? So if we can moderate the corona, we can enhance for betterment, better use of the nanoparticle in applications, right? So these are the kind of paper which published in PNAS, which was showing how this nanoparticle either coming through the pollution or accumulating inside the brain, causing the aggregation, and maybe was found in the Alzheimer patients. So it was associated that this nanoparticle can deteriorate the condition which can lead to amyloidosis disease. But again, there are certain publications which shows, right, like this kind of fibrillation can also be ameliorated if you can play with the nanoparticle interface. So how we can choose which interface we can try for the biomedical applications. So this is my current group, where we are focusing over the nano formulation which have the antimicrobial activity against the parasitic microbes as well, anti-cancerous, but basically I'm going to talk about the aggregation protein amyloidosis problem which can be, right, affected or which can help with the nanoparticle use, right? So this is the insulin, bovine insulin. Bovine insulin doesn't fibrillate. You can see over here, this is the thioflavin T probe assay, which is the only probe to check in vitro the fibrillation, amyloidosis is happening or not. And you can see it's a straight line. That means bovine insulin doesn't fibrillate, but as soon as you add the zinc oxide nanoparticle with positive interface, actually zinc ion is used in the insulin formulation to stabilize the insulin dimeric or hexameric structure, right? That is the human insulin. So if zinc oxide nanoparticle you add, as per the concentration, lag phase is reduced from 44 hours to just few minutes. Means bovine insulin, which doesn't aggregate at all, now it is aggregating relatively very fast, right? It is aggregating very fast, and surprisingly, the interaction between the nanoparticle and zinc oxide nanoparticle interface and bovine insulin is very insignificant. The interaction in submillimole concentration means it's a non-specific interaction. It's not like a specific interaction which is going to change the structure of the bovine insulin. And the change is also not very drastic, right? But it was forming the kind of fibrils. This is the fibrillar morphology that you see in the TAM. And in presence without nanoparticle, there was just amorphous aggregate, right? And they were having, those fibrils were having the cytotoxic effect. So we gave the hypothesis like this non-specific interaction was enough to bring the slight conformational change in the bovine insulin, which was opening the structure for the self-aggregation, or we call it self-assembly of the protein into amyloid, right? So is it problematic, right, for the bovine insulin? Now we are checking with the human insulin also, but with bovine insulin, if we can moderate the zinc oxide nanoparticle surface, as I said, right, we did it with using tryptophan tyrosine, relatively the cytotoxicity as well as the propensity to enhance the amyloidosis propensity in insulin reduced drastically. It was only forming the amorphous aggregate, not fibril. Just like, and that is in pH 7.4. Insulin, whether it's a bovine or human, they are only soluble in the acidic condition. No, they have not been checked in physiological condition, but where that is the condition where they are active, right? So in physiological condition, they fibrillate in presence of nanoparticle, but if we can moderate the nanoparticle interface, they don't. Now this is the second story. That is bovine insulin or insulin is a tertiary structure, right? That means they have a core which doesn't open up very easily because they are entropically driven, and burst phase is driving that process to get the tertiary structure. This is the story of intrinsically disordered protein, that is almost 30% of your proteome in a cell, that is made up of intrinsically disordered protein where the structure is slightly open up compared to tertiary or quaternary structure. And alpha-synuclein, most of people know who is on cell biology or so, because this is the causative protein for Parkinson's disease, right? And we have checked same nanoparticle, does it also enhance the fibrillation propensity in protein, in alpha-synuclein? And we saw, besides, instead of insulin, right, or the contrary to insulin, the alpha-synuclein interact with zinc oxide nanoparticle very strongly, very specifically. And that is not because of the interaction with the interface. That interface resulted in a corona of the protein, which made the protein accessible to self-assembly. And that assembly is mostly the entropically driven. That is entropy. There are two processes, right? The free energy process can be described whether it's enthalpically driven or entropically driven, right? And enthalpically driven, they are mostly the electrostatic interaction, ionic interaction, and so. Whereas in entropic driven process, they are mostly the hydrophobic interaction, water release structure, right? So that third step of the binding is the entropically driven process that is happening between the protein and protein, which is adsorbed over the nanoparticle surface. So we saw that in presence of zinc oxide nanoparticle, right, alpha-synuclein, which is without zinc oxide nanoparticle forming the fibril, they were forming the amorphous aggregate. And over here, it is just like an inclusion body, or you can say holding out process, where the aggregated protein is monomers present in aggregated protein is kind of physiologically active monomers, right? So they can be again used if you put in the system, they will act as a monomeric form, and by the depot effect they will release the protein very slowly. So it is not like one-way process that the agglomerate is formed, they can be dissociated back to the monomeric form, right? So we have seen the physiologically activities there, as well as there is no cytotoxicity if such agglomerate are being formed of alpha-synuclein in presence of the nanoparticle, right? So those were the two stories of IDP protein and tertiary protein. Now it is a story of Bacillus anthracis antigen, which we did with Professor Rakesh Bhatnagar in JNU, because he was having the BSL-3 facility. There we did everything. So we synthesized silver nanoparticle with negative interface, and silver nanoparticle is very cytotoxic because it has very high affinity for sulfur, and cysteine is the very catalytic site of a protein. If you engage it some other way, those proteins are non-functional, of no use, right? So we coated it with BLF. BLF is bovine lactoferrin, which is a cytocompatible protein, and that is acting as a corona around the nanoparticle, right? It was having very good affinity, very good structure we found, and it was internalizing inside the cell very easily. So, and this was having very less cytotoxicity, as well as ROS production was very little compared to when bare silver nanoparticle was used. And we coated it with the PA protein. PA protein prolom is only physiologically active in the monomeric form after the purification for 3 to 4 hours, right? At 4 degrees Celsius. But when we coated along this nanoparticle corona, that was physiologically active, or means they were having the immunogenic response inside the mice, when we could store it at 4 degrees Celsius for almost 4 to 7 days. Right? So you can see the shelf life of PA enhanced by almost manifold. And the immunogenic response, you can see this is the immunogenic response after the 42 days in mice of our formulation for PA. Means it was very good in creating the immune response. Now, so this was the graphical abstract we given to that journal. Corona was providing a surface multifunctional group where the absorption of PA was very strong. It was giving an agglomerate where all these PA were immunogenic, were behaving normally as a monomeric form, not an aggregated structure, right? It was inducing so this formulation was working as a very good adjuvant. But problem we started with, that was the model study or the preliminary study you can say. We started with the silver nanoparticle, which is not good for the biological application, even for adjuvant. So we tried our model system. We biologically synthesize aluminum nanoparticle, though aluminum microparticle or gel is nowadays used for adjuvant for PA. You can see when we use the biologically synthesized aluminum nanoparticle, PA absorption is almost 100% PA is absorbed. You can't see over the gel any free PA protein. And internalization was very good. The binding was relatively very strong compared to aluminum microparticle. That is all, again, the protein-protein interaction was predominantly governing the absorption, and the immunogenic response was very good, right? But where was the problem? Problem was at the 7th day. Compared to the traditionally or these days used adjuvant, our adjuvant, in our nanoformulation, there was one extra mice death on the 7th day. So what was the reason? It's not published yet. What was the reason? We dwell inside and we found that our nanoparticle probably is diffusing inside, and the protein which is being absorbed, we are taking as a corona from the host bacteria where we were synthesizing it, they were inducing the cytosolic pro-inflammatory response, right? And when there is a cytokine storm, because of that cytosolic storm, response, there may be a macrophagic response which one mice on the 7th day could not handle, and the extra mice death. That is our hypothesis, right? And that's that hypothesis because we see there is more like a pro-inflammatory response. Those cytokines are going up, right? So that was the thing. That was the thing, like our metal nanoparticle, especially the photocatalytic nanoparticle, can work in both ways. Can enhance the fibrillation or amyloidosis propensity, which can result in age-related disease, which will not be coming up as a symptom immediately. Maybe like right now we are checking in the Drosophila. In Drosophila, we are seeing the symptom within 10 days, right? But in human, it is not that fast, right? It may take 40 days to appear, right? So whereas if we moderate the interface of the nanoparticle, we can use it as a safety guard to stop or to ameliorate the protein which are prone to aggregation, like IAPP, like PA, like alpha-synuclein, and still have the physiological activity, right? So that's my idea, and that's the thing we are working on right now. Yeah, that's all from my side, from science point of view. And this is my previous group who have contributed for all this work. And these are my collaborators: Andrew from Yukon, Bhatnagar sir from JNU, and Mona Lisa Mishra and Rohan Dhiman. Rohan works over the Mycobacterium TV. And Mona Lisa ma'am working with the Drosophila system organism. So she is providing our the model for the study. And Salim from NICE and Sanjeev from AIIMS, with whom we are working, we are starting to work whatever result we get from the Drosophila over the, he is a neurosurgeon and he will provide us the feedback, right? So for NMR studies, we are relying on the Anirban, and yeah, that's all from my side. Yeah.
Thank you, sir.
Thank you.
Any one question? Okay, thank you, sir. We'll interact.
Thank you for that informative talk, sir. Our next speaker is Dr. Amal Kanti Bera. Dr. Bera pursued his PhD at the University of Delhi and his postdoctoral studies at the Einstein College of Medicine, New York, and the Southwestern Medical Center, Dallas, Texas. He also served as a visiting scientist at the Department of Neuroscience, University of Iowa. Currently, Dr. Bera is working as a professor at Department of Biotechnology, IIT Madras. His research areas include studying the structural and functional relationship of ion channels, focusing on neuronal diseases associated with receptors malfunctioning. I welcome you, sir.
Thank you. First of all, I would like to thank organizers for giving me chance to talk about my work. So I work in IIT Madras, and this is our campus. So if you have not visited our campus, you are most welcome to come and visit. So our campus is located inside a reserve forest, so you see all these animals all the time. Next slide, please. So the title of my talk, that from delicious to deadly, the dark side of trans fat. Next. So all of us like this kind of food, so-called junk food, tasty food, and each time we eat outside, we prefer like to have pizza, samosa, and so on. But you know that all these so-called junk foods are not good for our health. It has like lot of effects on our heart. Particularly, it can cause you heart attack if you eat it for a long time. It causes atherosclerosis and so on. So apart from high salt and sugar and oil, it has another composition which is called the trans fat. So what is this trans fat? In our body, all these fatty acids they have the double bond. If it is unsaturated fatty acids, they have double bond, and all these double bonds are in cis configuration. However, these trans fats are not synthesized in our body. It comes from our food, and they are the double bond in the trans configuration. So our enzyme doesn't recognize this trans configuration, and it cannot metabolize. So once this trans fat are introduced into our body, it stays there for a long time. Next. So it is known that trans fat can cause you cardiovascular problems, it can cause diabetes, and it can cause obesity. These are all well established. But we ask another question. So if you eat junk food, are you becoming more prone to depression, anxiety, or Parkinson's disease or other neurological disorders? You say no, because I don't see anybody is unhappy after taking pizza or pasta. You feel good. Your dopamine increases and you feel really good. Particularly kids, ask them whether you like pizza or dal roti. Obviously they will say pizza. Okay, so it gives you a good feeling, your dopamine is increased and you feel good. But what happens if you eat days after days? Are you becoming more prone to anxiety and depression? And so we ask this question, whether trans fat has any kind of effect on our cognition, on our learning and memory, on our decision making, and kind of anxiety, depression, and so on. Next. So we took zebrafish as a model system. So what we did, next please. So why zebrafish? It's easy to work, it is less expensive, and you can do lot of already developed models are there. They are very good model for neurological studies. So what we did, we took the zebrafish and we fed them with trans fat containing diet, and which is like eleidic acid. Eleidic acid is the most abundant trans fat present in the junk food, and then oleic acid, which is the cis fat. So we fed the zebrafish for about a month with containing different amount of trans fat or cis fat, and after that, after 30 days, we put them under stress. So what kind of stress? This is called unpredictable stress. So say for example, so all of you know that stress can cause you depression, right? But if this stress is predictable, suppose you know that you are going to retire, at the age of 65, so you are mentally prepared, you are not stressed. But if I say that tomorrow your job is over, it's a big stress. So that's why we give them unpredictable stress. So they don't know what stress is going to come. So suddenly we put them in hot water, hot means warm water, or cold water, or we put them outside for some time, and so on. So this kind of unpredictable stress, and the more important thing is that we put them in stress not in the same time. If it is in the same time, they predict that, okay, I am going to get some kind of treatment. So they get mentally prepared. So it is not stressful anymore. So you put them in unpredictable stress. So suddenly you put them in hot water, cold water, or put them outside, put them together, many fish together. So that if you continue for about 10-15 days, all these fish show the symptom of depression and anxiety. So we did that. We have 3 groups. One is eleidic acid fed group, which is the trans fat, and another is like good fat, which is oleic acid. And then we put them under stress, and then we did lot of behavioral studies. Next. So these are the kind of behavioral studies which essentially tells you whether they have the anxiety or not, they have the depression or not. So one is called the novel tank test. So in this test, what do? You take a new tank and put the fish there. If the fish is not depressed in normal condition, they will go to the bottom, they will roam around, they will explore, and then they will come up. But if the fish is depressed, they will come in one side and they will sit. They will stay there. They will not move much. Like, depressed person, they don't want to interact with others. Similarly, social interaction, as you know that if you are, not depressed, you are in a right mood, you will interact with your friends. But once you are depressed, you don't like to interact with others. So here also exactly same thing happens. Depressed fish don't interact with other fish, they stay away. Shoaling behavior, they have the tendency of swimming together. But if they are depressed, they don't like to do that. So all these tests we do the video recording and then analyze. And what the conclusion came, I'm not going to show you the data because of the lack of time. What we essentially found that TFF feed fish exhibited increased anxiety and depression-like behavior. So it is very, very obvious. You can, without doing any test, you can say that, okay, this group of fish is fed with the trans fat. So it is just a month of trans fat. They are all becoming, as it is, they look fine, they look happy. But the moment you put them under stress, they show the more depression and anxiety. So next we ask at the cellular level what is happening. Next. So what we found that trans fat is actually going into a membrane, all the plasma membrane, organelle membrane, trans fat is going and sitting there. So, and as I said, the trans fat has the double bond is in the trans configuration, which behaves like saturated fat. So membrane fluidity changes. If the membrane fluidity changes, whatever proteins are embedded there, it could be receptor, it could be ion channel, their properties are getting changed. So obviously, if you change the membrane properties, biophysical properties of the membrane, you are going to have the changes in the signaling process. So we looked at the different signaling process. Next. So first we look at the calcium signaling. Because calcium, as you know that calcium is responsible for your life and death. The moment the sperm fertilizes the egg, when the life starts, there is a spark of calcium, intracellular calcium increases. When the cells is dying, there is a rise of calcium. So calcium is very, very important for your life and death. And inside the cell, calcium level is maintained very low. It is about 100 nanomolar. If it goes beyond 1 micromolar, it becomes very toxic. So we looked at what is happening to the calcium signaling process once you treat the cells with trans fat. Next. Next. Yeah. So we looked at the release of calcium from the endoplasmic reticulum. As you know that endoplasmic reticulum is the store of calcium. So we activated GPCR because, as I told, that membrane fluidity changes. So the properties of the receptors is likely to be changed. So we activate the GPCR and looked at how the calcium is coming from ER to cytoplasm. So trans fat treated cells, they have huge rise of intracellular calcium. As I told you that increase of calcium too much inside the cell is not good. So trans fat treated cell shows a very high rise of intracellular calcium. Next. Then we looked at so when the endoplasmic reticulum release calcium, some part of the calcium goes to the mitochondria. So there is a crosstalk between endoplasmic reticulum and mitochondria. So we are looking at this calcium dynamics, what is happening once it is released from endoplasm, how it is entering mitochondria. So this is oleic acid treated, which is normal, good fat or cis fat. So calcium enters into the mitochondria and almost immediately, it comes back to the normal. So calcium inside the mitochondria, high calcium is not good because it can induce apoptosis. So mitochondria has the mechanism of efflux out calcium. So calcium goes out, but what happened to the eleidic acid? If you treat cells with trans fat, ER releases calcium, it goes to mitochondria and it stays there, it doesn't go out. So which is again a trigger for the apoptosis. So you can say that at the molecular level, at the organelle level, it is affecting our signaling process. Next. Next we look at the single molecule level, how it is affecting the behavior of single molecule. Our model system is different ion channels. Because as you know that ion channels are embedded in the membrane and you are changing the fluidity of the membrane. So they are likely to affect the channel properties. So we looked at a channel which is called the VDAC or voltage dependent anion channel. This channel is present outside the mitochondrial membrane, in the outer membrane. So we isolated this VDAC, and this VDAC is associated with apoptosis. Okay. So we take this membrane, we take this protein out, purify this protein, we make a synthetic bilayer, we incorporate this protein inside, and then looking at their electrical properties. So what we see here, if you have elaidic acid, which is the trans fat, the channel remains open much longer time. It doesn't close. It open, but it stays open for longer time. Whereas, if you have oleic acid, the channels fluctuate, opens and close. So now we also found the mechanism. So what happens? This VDAC channel has a alpha, it's a beta barrel channel. It is embedded in the outer mitochondrial membrane, and the N terminus is alpha helix. That alpha helix actually goes inside the pore, then the channel closes. When it goes out, the channel opens. The trans fat actually interacts with the N terminus, and it keeps it open for the longer time. So mobility of the N terminus of the VDAC channel is getting affected, and thereby it is keeping the channel for longer time. Next. So then we ask the question, if somebody is taking lot of junk food years after years, are you becoming more prone to neurodegenerative disease or not? The question, the answer is yes. I'm going to show you one protein which my previous speaker told already, alpha-synuclein. Alpha-synuclein, as you know, that it has the tendency of getting aggregated. And in Parkinson's disease, most of the problems is because of this aggregated alpha-synuclein. And one of the properties of this aggregated alpha-synuclein is that they forms channel in the neuronal membrane. So we purified alpha-synuclein, and then we put them in the synthetic lipid bilayer and looking at their properties in presence of trans fat and in presence of cis fat. What we found that if you have trans fat in your system, this channel remains open much longer time. Again, it is an indication that if this alpha-synuclein channel remains open for longer time, it is going to kill the cells. So taking all together, it clearly suggests that if you have trans fat, you are taking junk food for years after years for longer time, you are going to have lot of diseases. It can affect your learning and memory, which we didn't show here. It can cause you more depression when you are under stress. So the take home is say no to trans fat. That's all. And next slide, please. This is part of my group. I have so the trans fat work was, the alpha-synuclein work was done by Sucheta, Debanjan did VDAC, Dhriti did calcium signaling, Kalai did behavioral study. They are the current student, and I have, in one slide I couldn't show everybody. So I have a big team of people, and my collaborator, Dr. Prasun from AIIMS. And thank you for listening my talk. I'm ready to answer. You have any question?
Thank you, Professor Bera. First of all, thank you very much for finishing your talk in due time.
Okay.
So we will take a couple of questions only from students, because now we have program, we have to end up by 12. So we are just minimizing the time. Any quick question from the students, please? No question from student? Okay. One, maybe. You have question?
Actually, this is a very interesting study on the trans fats and the neurological action on this. These ion channels which are opened up, have you done some physiologic studies in this? Using Osing's chamber or a patch clamp?
Yeah, we do. These are ion channel data are like bilayer. We have a lot of patch clamp data also, which I didn't show. Yes, we do see the VDAC activity, which you see that it's tremendously affected. Other ion channels like HARC channel in cardiac cell, as you know that, HARC channel interacts with many drugs. This HARC potassium channel in cardiomyocyte, it is highly affected by the trans fat. So we did the patch clamp study, and we have our lab is patch clamp lab. We do patch clamp all the time.
I asked this question because these channels could be modulated with many small molecules,
yeah,
which are already in the market.
Yeah,
so you could have something to really ameliorate it.
Yeah, basically any drug you develop, you need to see whether it is interacting with HARC or not. So we checked HARC activity, it is highly affected by trans fat.
Okay, any question from student side? Okay, no question. So our next Speaker is Professor Nanda Gopal Sahu. He is professor in Kumaun University, expert in nanoscience, nanotechnology, and also he is expert in the waste management, polymer recycling. He has been awarded by many awards. So, Professor Sahu, please. Okay, then you take time 2 minutes. I request Professor Chafu Chou from Academia Sinica, Taiwan, who is the convener of this conference from Taiwan side. And I request him to address the gathering for BioHill 2023. Please. Please welcome Professor Chafu. Maybe by you can slide, maybe no need.
Okay. Okay. Good morning, everyone. First of all, I'd like to thank Professor Narayan Mishra for organizing this conference with some minor also contributed effort from our side. Basically, we're from Academia Sinica, Taiwan, basically based in Taipei. We are the topmost national research institution. And within Academia Sinica, we also have Taiwan International Graduate Program, which we set up a booth over there. Some of you may have already visited the booth. If you haven't, feel free to visit our Taiwan International Graduate Program booth within the Academia Sinica, which we provided 13 multidisciplinary PhD programs, okay? These are all multidisciplinary, so it's different from the traditional departments, or graduate schools in the universities. But we do work with top Taiwanese national universities to run this program in order to grant the PhD degree. So in this program, you will be able to explore. Oh, okay. Yeah, so basically, you will be able to, this is the Taiwan International College. We have 13 PhD programs. And then we also have, basically, it's all scholarship-based PhD programs, okay? So next, please. So, can you click on that? Yeah, so PhD program, we have 13 multidisciplinaries. So we have 10 partner universities, and it's all English environment, okay? And we have life sciences, mathematical, physical sciences. So these are the life sciences programs, okay? And next one. Next slide, please. So basically, for example, this one is artificial intelligence, Internet of Things, This is a very new program in the mathematics and physical sciences. So I'm also the, sorry, can you go back to go back to the previous slide? The previous slide. This one. Yeah, thanks. Yeah, so I'm currently also serving as coordinator of the nanoscience and technology program. But in the next within probably 2 years, we are trying to expand the nanoscience and technology program into the, including the quantum science, okay? That means, quantum computation and quantum computers. So we have already almost 600 students from 47 countries as of this year, okay? And then our graduate also had, work at different fields. The next slide, please. So so far we have 651 PhD graduate and published 2,100 something SCI publications, and also 70% of our alumni are working in university research organizations, and also 30% are working in industry, particularly semiconductor and pharmaceutical industry. And in fact, some of our graduate Indian students, they did work in TSMC, Taiwan Semiconductor Company, okay, the largest semiconductor foundry in the world, right? And these are some of your Indian and also some of our domestic alumni. So, for example, Miss Dr. Sanapati, she's a postdoc working in Oxford, Oxford University, and Sachit is now a researcher in Switzerland, okay, and Haider is now research staff in Czech Academy of Sciences. And one of our alumni, she is a Taiwanese, but she's now working as assistant research fellow. It's like assistant professor in the Institute of Plant and Microbiology. Okay. This is our kind of a financial support of the fellowship program. So we offer about US dollar $1,100 per month for the fellowship, and we have some research performance fellowship, for the senior students who make good progress. We also have presidential fellowship, this is about 50K Taiwan dollars per month. That's about 1650 US dollars per month, okay? And I also offer TIGP Dome for the PhD students coming and to our campus. So this is like a single room, but we also have like a family room. So you have a private bath and with a very affordable rent, for students. Okay, next please. So we also provide a travel grant for students to go to the international conferences. Okay. Which including all the relevant expenses for the conference. And also, since 2006, we have already 185 students received these awards. Okay, next please. So this environment, because Academia Sinica is like the National Academy of Sciences in Taiwan. So we have lots of the domestic academy members and also foreign associate academy members. They are able to interact with students, so they all of them, there are like more than 10 Nobel Prize winners, often give colloquium or special seminars on campus. Okay, next please. Yeah, okay, I think we can skip this. Basically, the idea is this is, we also, in conjunction with some international organizations like UC Davis on the biopharmaceutical program, and also the European Molecular Bio Organization for thesis writing and paper publishing work. Overall, we offer a very multidisciplinary, multicultural environment. So for students studying this program, basically you enjoy the joint merits, okay, of multicultural and multidisciplinary environment. Okay, thanks. And these are also the different kind of glimpse of the program, like a biodiversity. We have the research boat. You can go out to some islands, around Taiwan, to study the biodiversity of even the marine biology or coral reef status. Okay, next please. And also we have like a career build-up activities. Yeah, next please. And also job fairs. We encourage students to attend job fairs also. So life in TIGP, lots of events, including the Lunar New Year, okay, celebrations, and Dragon Boat Festival, mostly in June, And also this pottery making workshop, right, in one of the small town, like a pottery town in Taiwan, Yingge. Okay, yeah, please. And this is how the facilities on campus. Okay, so it's pretty well equipped. Next, please. And counseling center, TIGP Dome. And these are the, and also living in Taiwan or studying in Taiwan is very convenient. Almost every street corner you find a good convenience store. So, okay, they offer everything. Almost your 3 meals can be done there. Okay, and also all the banking, whatever, it can be done in the convenience store as well. And Nangang, the location of Academia Sinica is in Nangang. Nangang is the starting point of the Taiwan high-speed rail. Okay, the first station from Nangang, only about 5 minutes from our campus. Okay, next please. And also the software industrial park in Nangang is very close by, also 5 minutes walk. So I think how to apply, we don't need to go to the detail. If you have any question, you can ask Flora. She's here to serve you. Okay, yeah, she's sitting there. Okay, and then normally our application is in deadline is the end of January. We have just finished this year's evaluation. Of course, we are receiving quite a lot of applications from India. And also, we already granted a lot of applications to Indian students also. So we welcome you for the younger generations. You can consider this program. For the faculty members, we also will try to organize some bilateral workshop, between Taiwan and India, so that we can promote more collaboration, okay, to advance science all together. Okay, thank you for your attention. Thank you.
So, thank you very much, Professor Chapu. I request you to say about your expertise.
Oh, my expertise.
Biosensor, whatever.
Okay. Our lab, my research interest is on the microfluidics, nanofluidics, and using this platform for biosensor development. So we do make a tiny nanostructures like electron nano gap down to 5 nanometer. So we can use AC electric field to trap biomolecules like peptides, proteins, DNAs, and we can use that to study the conformational change, structural change, and also perform some sensing analysis using these devices. So, for example, using this nano gap to promote surface plasmon enhanced Raman scattering, right? And using very small number of molecules, could be down to single molecules, okay? And we do lots of DNA polymer dynamic study. And also we use take advantage of a special confinement, geometric confinement provided by the microfluidic or nanofluidic environment. So you can study bacteria, for example, how bacteria are being squeezed into a nanospace, which is much smaller than the diameter, right? And to study the proliferation and the cell division process when the bacteria become like a chapati, bacteria, like a pancake, bacteria, and they can still perform, the shape, morphology has been largely changed, but they can still perform a cell division, okay, and then to go, to recruit back to its wild type morphology if this geometric constraint is being released. So these are the different areas. So we do fundamental biophysics study, polymer physics, but we also do biosensors, also do some tissue engineering scaffold. One of the Indian students, he just graduated, Prachanda, now he's a postdoc in the in the lab. He's solely responsible for single jet, silkworm-like 3D, scaffold building, using a conventional electrospinning setup. But outcome is very exciting. So in fact, from the poster, you will see one of the images of the 3D scaffold is made by Bachanda. Okay, so if you have interest, you can talk to him a little more. I guess he will probably having some later poster presentation if it's still going on today. Okay, yeah. So that's all my brief introduction of Academia Sinica and also the TIGP program and also our research. So see if you have any questions you want to address. And I'm also very glad we have been having a long time collaboration with Professor Mishra, over the years, and he had been in Taiwan many times, Okay, he knows how to live even better than the local people in Taiwan to get himself around. Okay, yeah. So if no further questions, please address any questions about our Taiwan International Graduate Program to me or to Flora. And if you are interested to know a little more about our research, we can also talk privately. Okay. Thank you. Thank you. Thank you.
Okay, thank you, Professor Chapu, for giving valuable time and for being convener, also main sponsor of this conference. And really, we are grateful to you. And Professor Chapu, just one thing about him I want to tell. He is a vegan. He does not take milk. So no this dahi, no milk, no paneer. You see fit body. Every day 3 hours meditation. That he will do at any cost. So means very simple lifestyle, but working very high.
No,
so once again, thank you. Thanks to Professor.
Thank you. Appreciate. Yeah, thank you.
So our next speaker is Professor Nandagopal Sau. I already told he is professor in Kumaon University, and he is the professor in charge of nanoscience and nanotechnology program in Kumaon University. He has several publications, patents in the area of nanoscience, nanotechnology, waste management, recycling, and we welcome Professor Sahu.
Thank you. Hello.
Please.
Thank you. Good morning to all. And thank you, Professor Mishra, Professor Vishal, and Professor Sogoto Hazra, and other organizing committee to invite me to present some of my work. Yeah, basically, nowadays actually I'm working on the waste management. So today actually I'm going to show you some of my results. There is a waste to wealth. I know everybody knows what is the waste, so I'm not going to discuss about what is the waste, but how to handle it and how to upcycle the waste and how we can add the value to the waste. That is I'm going to show you. And that is actually upcycling of waste material to carbon nanomaterials for various applications. So nowadays, there is two categories of the waste. There is the biodegradable and non-biodegradable. And basically, the non-biodegradable is the polymeric waste. And this polymeric waste, somewhere actually you can see the dam, the plastics. And, if we can dam the plastics, what is the problem will be arise from the plastics. If we can burn openly, there is a different of the toxic gases will be involved. And that will be the pollute the environment. And also, the plastics can choke the drain, and during the monsoon, that is called the flood. And not only that, there is no actually organization are there to handle the plastics, but there is the most of the disadvantage of the plastics, but you know, because of the plastics, our everyday life is very easy because of the plastics can be used in everywhere. And because of the durability and low cost, so plastics can be used in different fields, the packaging, textile, electronics, agriculture, infrastructure, everywhere you can use the plastics. But problem is that when we can throw the plastics here and there, that is the problem. And already, you know, the government also banned the single-use plastics. But still, you can see that every year, the plastic production near about 10 to 11% increasing because of the use of the plastics. Next. So you can see this is the, you know, the plastic in the lake area of Nainital. There are a lot of plastics, and that the pollute the environment. And when you can throw the plastics into sea and rivers everywhere, just you can see the sea fish and other animals can eat as a food and just like the stomach and they die. So basically, we can destroy our environment. Next. And recently, actually, you can see that in the Faridabad, this 2011, the near about 71 kg of plastic waste came from the cows, and I think both the cows and the baby died. So near about 5 million cows roam in the city. So if we can throw the plastics, basically we can destroy our ecology. And also recently, you know, the carbon-carbon linkage. So why not we can use this carbon and we can develop something new materials. So that's why our first objective is how to synthesize the reduced graphene oxide from waste materials, and this graphene oxide can be used for the various applications. And basically, what is the graphene? Graphene is a single sheet of graphite. There is the sp2 hybridized, there is a honeycomb structure, and there is a mechanical and thermal properties very high, and also conductivity is very high. And this graphene actually discovered in 2004 by Professor James and Novelso, and they got the Nobel Prize in 2010. But the problem is that the mass production of the graphene and cost. They have very high cost of graphene. That's why it is very limited application in the industry. So when we can start actually in 2016, actually we started in my lab is from the zero. So in 2016, that time we did not have any fund. So we bought actually the one reactor from the market, only spent of the 5,000 rupees, and where actually we started this work. And next, actually, we have developed the waste recycling machine, where we got actually fund near about 2.24 crore from the ministry, and go. And this is actually waste recycling machine where we can install in the Nanoscience Nanotechnology Center, Kuma University. And okay, this is actually the shredder where we can crush the plastic in the very small pieces. Next, that is the washing machine where you can wash the plastics. And next one, this is the hot dryer where we can dry the plastics. And next is the mixing unit where you can mix the plastics with catalyst. And that is actually important step. What type of catalyst you can use and what is the percentage, and also that depends on the different parameters. And next unit is the pyrolysis unit where we can load the plastics. And that is first we can start the machine with a diesel engine. And when the heat up to the 200 degrees centigrade, there is a different type of gases will be evolved. And these gases come out, you can see that there is a yellow pipelines, and this you can recycle again the toxic gases into the main chamber, and that time you can stop the machine. These gases can run the whole instrument. This is the actually beauty of our machine, and that's why no toxic gases come out the environment. I will show you the video. Yeah, this is actually the whole process, and this is actually we have already received the patent. Next. This is video. Click. Yeah, yeah. This is actually our nanoscience nano center and Combined University, where we have installed this waste recycling machine. Yep. Yeah, very slow. Yeah. You can see my students actually is working on the plastics, and that is actually for the demos. Just we can use the PET bottles, and you can see the very small pieces of the plastics. This is the rectangular shape. And actually, it depends on the kata units where you can get the plastics, very small pieces, and we can get the final product. Yeah, this is, you can see the very small pieces, and after that, that is the washing. One. Yeah, this is the washing, and next, you know, this is the drying unit, where you can dry the plastics. I think you can quick, that's okay. Yeah, here actually we can mix with the catalyst. Actually, we have developed our own catalyst. And after mixing, we can use in the chamber, main pyrolysis chamber. Yeah, this is 100 kg plastics capacities. And then actually we run whole experiments and presence of nitrogen, inert gas, and we have a diesel engine where you can start the machine, and last we can get heat up to 400 to 500 degrees centigrade, and then we can get the raw graphene. And another is, please show the.
We don't have time, please. I request just. Please, you will have just last slide.
Yeah, okay, that is video.
Video.
Just,
go to the last slide.
You can go. Yeah, this is actually we have transferred to the technology transferred to the industry. Already one industry and another two industry we are going to transfer the technologies. Already commercialized this technology, and because of this technology, actually I received also the national award. Go.
Just last
yeah,
slide.
This is actually the main importance, the production cost and the product cost. You can see if we can run the 100 kg plastics, just the cost of this is only the 10,000 rupees. And we are getting near about 15 to 20 kg of graphene. So the graphene cost reduced up to 1,000 rupees per kg. I think you can go last. Whatever I can say. Okay, this
sorry,
is I can finish my
Professor,
talk.
sorry.
Yeah, actually I have a lot of work.
Actually, people will learn once they know your name, your area. People will study.
Okay.
Research paper.
Yeah, this is actually the I also.
Slide without slide you will go. Nah, there is some problem.
Just slide.
Yes.
Yeah, this is my groups, my students. Actually, because of my students, we have developed this technology. This is the hill area. No, Italy, the hill area.
Thank you. So our next speaker is Professor Kunal Bhattacharya from Patanjali Research Institute. He is expert in nanoscience and nanotechnology and application of Ayurvedic medicines through nano carriers. Professor Bhattacharya, please. अरे भाई सारे टाइम व्हाट्सएप देख रहा है. Okay.
Thank you, sir. So I would start by thanking the organizers for giving me the opportunity to present our research that is ongoing in the Patanjali Research Institute. And I would also like to wish everybody a happy Bengali New Year. So I start with my work. That one of the work that is ongoing here is to apply one of the iron bhasma particles for treating anemia, that is iron deficient anemia. And the problem with that is that it has a very low efficacy because it is coming from a serendipitous research. So what we are doing is that we are transforming that into nanomaterials and we are trying to increase its efficacy. So I'll present that work with you. So I'll skip the first slide because this is a nanoscience conference, so everybody knows what it is. So as per the US FDA and NIH guidelines, the daily uptake of iron in people, by normal people should be between 7 milligrams to 15 milligrams. And it goes up for during the time of pregnancy and lactation. However, when the uptake of iron is not adequate, it leads to iron deficient anemia. And it's very concerning that in India, almost 58% of the children and 53% of the non-pregnant and 50% of the pregnant women are anemic. So this is the basic outline how the iron is actually absorbed inside the body. If you look at this, this is the small intestinal region, and these are the enterocytic layers. There are a layer of enterocytes which have formed the microvilli. So when we are delivering the iron, we are delivering it either in a form of heme iron, which has its own heme transporter proteins, which goes and gets collected into the ferritin inside the enterocytes. This is the protein that is reason for the storage of the iron. Other way around, back please, sorry. Other way around is to give the iron 3 that goes through the reductase process to transform into iron 2, bind with DMT1, that is present on the surface of the enterocytes and go inside the enterocytes and bind with the ferritin protein. So the reason why the iron is always bound is that one thing that you have to realize is that iron is more like you can say a shopaholic. That is, a free iron is very keen on oxidizing the biomolecules that are present around it. So it always is in a bound form with the proteins. So what we are using here is an iron ash that was produced by the Ayurvedic process, which requires a high temperature incineration of ferric oxide that has been aged for 50 or more years and transformed into Fe2O3 or hematite. And this is then treated with different type of decoction, herbal decoction for oxidation reduction reactions. And finally, you have the iron oxide particles that is used for the treatment of anemia. So the first thing that we did was we reduced the size of the iron by physically grinding it down. And then what we did was we coated the iron with ascorbic acid. Why? Because vitamin C or ascorbic acid is capable of transforming the iron into iron 2, and that is bypassing the iron 3 reduction process and directly taken up by the enterocytes. So the first thing was to look at the ratio of iron to ascorbic acid that we needed. And as you can see, the 4:1 ratio was the best one for making the iron bioavailable at pH 7 and pH 10. These are the small intestinal state, the pH state of small intestine. The second thing that we did was to standardize the time that is required for the coating of the iron nanoparticles. And we could see that by 24 hours, we had the best release of iron from the iron particles. So then we went for a dynamic light scattering technique analysis to see what was the actual physicochemical properties of iron. And as we can see that this was the original iron particles that you can say that was around 1 micrometer in size, which was grinded down to 100 nanometers. And then when it was coated, it went up to 120 nanometers. When we did an FTIR analysis, we can see that the groups, the functional groups that are present on the surface of the iron and the ascorbic acid were actually present in the final product, that is the ascorbic acid capped iron particles. And using an SEM, we could easily see that the iron particle in its original shape was present as clusters that you can see here. And when the grinder happened and the ascorbic acid capping happened, it reduced down up to 100 nanometers. But there was no change in the elemental composition, so we could validate that we were not changing the chemical composition of the particle per se. Then what we did was to take it to the physiological condition and see, does the particle behave what we really want it to behave as? So this was the raw nanoparticle, and the pH of the stomach is pH 2, and the intestinal pH can go between 7 and 10. So you can see that the raw particles were releasing iron at the pH 2, which is very bad because there it can do a Fenton type reaction and can release a lot of free radicals that can have a damaging effect on the stomach lining. However, when we went for our own product, so we could see that the particles stopped releasing iron at pH 2, but it was increasing the releasing iron 2 specifically, at a significant amount at pH 10 and 7. So, was the coating responsible for that? That answer came from an XRD analysis where you could see that these two specific peaks have vanished off when we had the coating on the surface because of the higher intensity coming from the blocking coming from the ascorbic acid. However, when we went to pH 10 treatment for 24 hours, these two peaks came back, showing that the top surface layer of the nanoparticles were removed at pH 10, and that was capable of delivering the bioavailable iron 2. Then, going towards more physiological conditions, we used an enzymatic system. This is what we call a simulated enzymatic system, and this comes from a paper that was first published by my Harvard group, and now we are using it in our Patanjali Research Institute. This has 3 phases: the mouth phase, the stomach phase, and the intestine phase. In the mouth phase, you have different type of enzymes like amylase. In the intestine phase, you have the acids and the like the H2O2, H2SO4, and important salts. And in the pH 10, you have lipases, specifically pancreatic lipases and bile salts. So when we did that experiment, we could see that we were capable of blocking the release of iron in this stomach phase, which was actually at pH 2. But when we went for the small intestinal phase, in the presence of the enzymes, we started releasing iron. Inside the intestine, a food can stay between 2 and 6 hours before it is taken out from the body. So what we did was, we further extended the time from 2 hours to 6 hours, and as you can see, by 6 hours, we reached a significant release of irons from the same nanoparticles that was not working. Now the question arises, is it bioavailable or is it the nanoparticle that has been taken up? So what we did was, we took the human colon epithelial cells, or CACO2s, and we fed them with the iron particles for a period of 24 hours. Later on, we looked with an ICP-MS in the particles, in the cells, and we could see there was a significant increase at the lower concentration of the transformed nanoparticle as compared to the raw material. That was around 100 nanometers. But there was no change in the bioavailability of the iron, the biocompatibility of the iron. As you can see, the cells were completely viable. So finally, when the bioavailable iron is there, is it getting stored inside the CACO2 cells? So what we did was, we looked at the ferritin storage protein content. And as you can see, when the cells were fed with the ascorbic acid capped nanoparticles, we had a significant increase in the ferritin content inside the cells, which was showing that our iron availability inside the CACO2s was good, and the free iron was actually capable of binding with the ferritin and becoming storage proteins for later bioavailability. So finally, from this study, we could conclude that it is capable, we can transform the iron-based particles that are available inside the Ayurvedic formulations into nanoforms and transform them into surface to make the bioavailability of the iron more, and more specifically in iron 2 form, which is easily taken up by the enterocytes. And this we could calibrate based on the pKa value of the ascorbic acid to only open up in the alkaline conditions and not in the acidic conditions. The final thing was that these were highly biocompatible and were capable of being stored inside the enterocytes in the ferritin protein content. So this is the preliminary study that we have done. And finally, we are going towards more sophisticated models, that is using transfill models of the enterocytic layer, where we will look at how the iron is actually processed inside the cells and getting released out into systemic circulation. Thank you.
Thank you, Professor Kunal, for giving your valuable time and for the wonderful talk. Any question from the students? Students, no कोई सर को दो सर question है? So no question. Also we have limited time, so
one question. That there is a preparation with iron fumarate with a soya lipid, which is released at pH 4 in the stomach and absorbed exactly how you will need. So the pH 10 is a non-physiological, and you need to really look at two things. Firstly, that whether there is any Haber-Weiss reaction, free radical generation with your particle, and secondly, that they should not raise ferritin content by iron. Stored iron is detrimental. So that is why in thalassemia and other way which stores iron, we give the chelators. So these are the few just suggestions to you.
okay. Once again, thank you to Professor Kunal Bhattacharya. Our next speaker is Professor Chandan Kumar from Central University of Rajasthan. He has expertise in nanobiotechnology, piezoelectric energy harvesting devices, and flexible electronics. Let us welcome Professor Chandan Kumar.
Yeah, thank you, sir. Yeah. Good morning, one and all. First of all, I'd like to thank you, sir, for organizing, and his team for organizing such a wonderful conference at such a wonderful place. Hi, I am Dr. Chandan Kumar. Currently, I am working in Central University of Rajasthan. So I will be giving my talk on piezoelectric energy harvester for low power energy sensors. Next slide. So what is energy harvesting? Let me introduce first quickly. Energy harvesting is something like harvesting. We all know this term comes with generally with agriculture. So harvesting is something we reap or we sow something. So energy, in the same way, energy harvesting is generally, we know that energy is neither created nor destroyed. So in energy harvesting, what we do, the utilized energy or the energy which is going through, we are just able to try to take some part of that energy and utilize for our different type of work. There are different type of energies are there. We know wind, water, solar. These are very large, comes under large section. And in micro, the temperature, vibration are there, mechanical energy, which we try to harvest. So presently, the current world scenario is that we know that our energy dependency is mainly on coal, petroleum, natural gas, nuclear. Because of this, we know that in many wars are also happening because of this energy crisis only. And some other part is 2.9. So our focus is there, 2.9. We try to maximize that 2.9 energy, that is the non-conventional source of energy, which to make it to the 100% or to make it used to our human beings. We know that, just a moment, the conventional source, that is the petroleum, coal and all, they are mainly contribute to pollutions, global warming and rising fuel prices also and others problem also. So on the other hand, generally human beings do a lot of work mechanically. Inside our body also many organs are continuously working. So we try to capture those energy that is basically goes on the environment as a waste energy. We try to capture that some part of energy and make it useful. So even if we harvest one small portion of energy, it will be very beneficial for our meeting energy demands. So some common energy harvesting sources are thermals, thermal, mechanical, electromagnetic light, natural and all. What we do, our focus is on vibration and stress. So yeah, this is our methodology and all, how we work. We work on a basic chemical, we choose some materials. Basically, our material is piezoelectric polymer. We all know that what is piezoelectricity. Piezoelectricity is something like, it is a material when upon mechanical impression or mechanical pressure, it gives you charge. Charge separation happens on the surface, and through some circuitry, you can draw that energy in the form of current or voltage, and then give power to some your devices. So this we do it through normal chemistry, like basic solution route method. We mix some materials like different fillers and all to our polymers. Our base material is polymer and its copolymer, PVDF and its copolymer. We mix different type of fillers in it. Fillers, why we mix? Generally, PVDF is an insulating polymer. That is, it doesn't contain any charge and all. So we fill some fillers or electroactive material to make it a channel or make the charge flow inside it. Then this is our, through molding solution and all, we make materials of micron size, okay? Micron in the range. This is our polymer. Through this type of device, we are able to power till now. We are able to generate power till 28 microwatt, which is enough to supply the natural pacemakers and various biomedical devices like hearing aids and all. Yeah. So these all are characterization. I hope everyone know that. After developing materials, we just check whether the materials which we have made is expected or not. Okay. These are some results and all, various SEM techniques and XRD and all, just to confirm, like generally what happened in polymer and all, for making it as electroactive polymer, beta phase is very necessary, beta and gamma phase. That is because these phases are responsible for the electroactive polymer. So basically in base, it is in alpha phase, as you can see. This is a normal alpha phase. So upon addition, we just check whether beta phase or gamma phase occur there or not. So this is the validation of different techniques, characterization we do. Yeah, how we measure electromechanical response. So after the polymer or the device is being made, what we do, we just check we just quantize the voltage or charge which is able, we are drawing. So how we do basically like normal biomedical motions or biomechanical motions like talking, speech, then finger tapping, through movement, leg, folding of hands and all, we place our device like that and we can use there. So through normal finger tapping, this is the till 20 volt we are able to achieve. This is the 3D representation of bending, twisting, walking, foot tapping and finger pressing. So as you can see, up to 32 microwatt per centimeter square, we are able to produce the energy. Yeah, this is the through bending, twisting and all. Also these energy we are able to store in capacitor to make it use for the later use. through walking, charging and all. These are the show like after making the device, we draw it from appending it with electrodes and through some electronics circuitry and all. So this is one circuitry, such a basic circuitry like a full wave rectifier, through which we draw the, as you can see, we are able to glow some LEDs and all. Power is being driven by just normal finger pressing. So some videos are there. First one. First one, you can minimize and then play. First one, the top one. No. Okay, fine. Some issue will be there. So in that I have shown that how we are charging or we are giving power to different biomedical sensors and device. And here we can show that we are charging capacitor. That power can be later used like power bank and all. Yesterday many of us visited for rafting and all. We have gone for camping and all. So might someone have faced the issue of their low mobile phone discharge and all, battery and all. So we are now in the process to solving, like we can store the energy from our device and we can use later for the power of our, to power the small mobile phones and a smartwatch or the laptops and all. Yeah. So this maker, hearing aids and all. Now we are currently working on to power the some wireless sensors and all. Wireless sensors and all, sometimes it is very hard to go and repair some damaged wireless sensor or to power to change the battery in that wireless sensor. Or sometimes the position or location of this sensors are such that it is very cumbersome to go and change. So through this energy harvesting without having any battery and all, we can continuously give the power to that sensors and all. And our main focus and target is on to solve the problem of the common people like cell phone, laptops and music player, to power the music players and all. And through body movements also we have shown. These are the macro power energy harvesting which is already going on. Yeah, the design challenges. It is not so easy as it is look like. These are some design challenges like first of all, whatever we are making, the efficiency is very low. Why it is low? That for power we know that voltage and current is needed. Voltage we are able to generate up to 150 volt, that is fine. But the current range is very low. Why? Because the basic polymer which we have choose is insulating polymer. So it is some limitation is there that we can't go beyond level. But continuously we are going through it and we have found some positive results also. And power is subsequently increased from nano range to micro range, which is sufficient. Also, yeah, thank you.
So thank you, Professor Chandan, for wonderful talks and a lot of new information for energy harvesting. So any question from the students, please? Very fast. No question. Then we start the next talk.
Thank you.
Next speaker is our Professor Sandeep Chaudhary from Central University of Rajasthan. He is expert in biosensor, environmental engineering, and also healthcare diagnostics. So let us welcome Professor Sandeep.
Hello. Yeah. Thank you, sir, for providing me this opportunity. And thank you BioHeal 2023 for organizing such a nice event. Myself, Sandeep Chaudhary, I am currently working as Assistant Professor at Central University of Rajasthan. So today's my topic of presentation is biocompatible fluorescent biosensor for food quality monitoring. So if we come, yeah, can you please next slide? If we go with the term that is biosensor, so biosensor is an analytical device which convert one form of energy into another. If we take an example, so enzyme can sense a particular analyte like urea can be sensed by urease and convert in the form of another energy. And that is sensed by the photo transducer, and then the signal can be converted into the digital form for further signal processing. So, the term fluorescent biosensors start with the process of fluorescence, which is a molecular phenomena in which a substance emits light upon light excitation. And this is governed by the fluorophore, that is further classified as organic, inorganic and biological fluorophores. So currently we are targeting the carbon quantum dot based biosensors. So we have synthesized this carbon quantum dot using phthalic acid and triethylene diamine TED dissolved in DI water, microwave radiation of 1 minute, and then sonicate, filterize and lyophilize to get a powdered carbon quantum dot. And then we use the pH property of this carbon quantum dot for further signal processing. So before that we have characterized this, and the TEM images are showing that these are the size is around 5 plus minus 2 nanometers, which is less than 10 nanometer. And we perform one interesting study here is that carbon quantum dot is photostable when is irradiated to a UV LEDs. So this shows that as compared to a natural or the fluorescent dye, carbon quantum dots are quite photostable in nature. Next slide please. So we use this carbon quantum dot based biosensor for studying the milk spoilage. As we all know, milk is consumed by all age of people, and currently many of you are consuming milk in the form of tea. So this milk is with the time goes, this milk pH is decreasing. Why? Because lactose is converted into lactic acid. So pH is decreasing. And our carbon quantum dots are also pH sensitive. So with the help of carbon quantum dot, we predict the pH of milk. And it is useful for the common man to predict what is the age of this milk. Either go for the paneer or chai or dahi. So this prediction can be done at this place. So next slide please. So yes, also many of the vendors do the milk adulteration study, milk adulteration practice, in which they adulterate the milk with urea, and that is very harmful if we consume the urea adulterate milk. So adulterate in milk, then its pH is not changing, and it is very harmful. So people cannot detect, a pH meter cannot detect the spoilage of the milk. So our biosensor enzyme based carbon quantum dots are able to predict this spoilage, this adulteration of urea as well. So how? This is a schematic representation of overall setup. The carbon quantum dots are mixed with the milk sample, and the readings takes place on the fiber optic spectrophotometers. So first we perform the pH study, in which we can see that from 3 to 7 pH is increasing, the intensity of carbon quantum dot is also increasing. And when the urea level is increasing, so 11 millimolar is the unit, is the limit that is set by the FDA. That is a permittable limit of urea that is present in the milk. Above that limit, it is adulterate milk. So we perform a spike in recovery studies, in which found that our percentage recovery is approximately 99.2%. So also we perform this spike in recovery equivalent for urea prediction in milk sample. So this is the overall conclusion. We are able to predict the spoilage and adulteration of milk sample using the carbon quantum dot based biosensor. These are biocompatible, and these are also not harmful for the human body. So this is the overall thing. Thank you very much for your patient listening. Thank you.
So now I request Mr. Piyush Padmanabhan. He is the CEO of NBIL. So your slide is ready? Yeah. So we welcome Mr. Piyush Padmanabhan. He is the CEO of NBIL, next big innovation limited, their interesting product is 3D bioprinter, by which we can print any type of scaffold which can be used for tissue engineering and regenerative medicine. They made the Make in India 3D bioprinter, which is much more less cost compared to the other 3D printer which are US based or other country based. So we welcome Mr. Piyush to introduce his product, and maybe I request what could be the cost, please inform so that we can contact you for the order. Please, Mr. Piyush. And those who wants to go to Dev-Prayag, here is the agent available. You have to submit rupees 500 and register your name. So it will, lunch will be from 12 to 1, and then by 1:15 we will start from the car parking of Paramarth Niketan to Dev-Prayag, and we will come back by 7 or 7:30, made from 8 to 9 we will have lunch. Also same time there will be poster session, and then from 9 to 10 we will have some cultural function as well as award distribution ceremony, and then maybe some, all those who are interested, we can dance together to end the BioHealth 2023 for today. But tomorrow there will be our holistic tour to the near waterfalls. There also we will enjoy, and we will have breakfast on the way or here, and lunch also on the way. And so please register your name if you are interested to join Dev-Prayag. Here is the agent. So please register your names. And we have to finish our lunch by 1 o'clock. After 1 o'clock there will be no lunch. Please remember. Okay,
yes,
Mr. Piyush, please.
Yeah, I'll just wait for the slides. Hi everyone, I'm Piyush. I'm one of the founders of Next Big Innovation Labs. I'm going to keep this short and brief. It's not going to be promotional about the company, but rather about our journey and how we got into the field of bioprinting. So we basically work on two main concepts. We work on deep tech bioprinters, so we build those deep tech bioprinters. Not only that, we utilize these bioprinters to bioprint different tissues internally. We customize the tech so that different people across the globe can utilize it, and that the main focus is to keep it more as customized as possible. The next thing that we also focus on is, when we're looking at bioprinting, it's a very new field. People need to understand exactly what the technology is all about. And that's why we got Next Big Learning into the picture. So with Next Big Learning, the focus is to see how we can, kind of build out the knowledge base in India and globally around bioprinting. When we got into the field of bioprinting, the first thing that came into our mind is, okay, printing organs for transplantation. But I'd like to make a correction here. It's not exactly organs that you're printing, right? You're printing smaller tissues, microarchitectures that can go into human beings for transplants. So that is exactly what we are trying to do. The goal is that, the primary goal of any bioprinting company is that, to build organs or tissues for transplantation. But then as and when, we started out, we started speaking to a lot of researchers at that point of time. This is in 2016. So we were talking to a lot of them about how we could basically use bioprinting as a company. How could we grow as a company in the space of bioprinting? And that's where we basically went on to explore a lot of the other advantages of the technology. It was more on how precisely you could position cells. Micron level architecture, micron level precision when you're building such complex architectures is something that is really important. The ability to work with multiple cell types. types, where you're basically printing multiple cell types. You're printing with multiple cell types, layering them one on top of the other, and this allows for also better cell-to-cell interactions. But when you're looking at a more commercial perspective, right, the world is moving away from animal testing. Now, as the world is moving away from animal testing, you're looking at alternatives, and that is where you're looking at creating tissues at scale. So what we are looking at is building machines that can actually build these tissues at scale. They can go into cosmetic companies, pharmaceutical companies, they can go into chemical companies as alternatives to animal testing. So when we started out, we obviously knew that a lot of the researchers told us that, you need to build out your own technology. You shouldn't be focused on using someone else's tech. You need to create your own engineering ecosystem. So we got supported by a few grants from the Karnataka state government, from the Department of Biotechnology, from the Department of Science and Technology as well. And one of our corporate partners was Autodesk, that allowed us to utilize all the softwares to build out the engineering. Next slide. So this is some of our initial work that we did. So the first thing that we did was we worked with Majumdar Shaw Center for Translational Research, where basically, we were printing in these, the slide, what do you call it? The slide discs that you get. They're really tiny, so we were able to print within that. Those were the first prints that we did. Next. The next one that we printed was with a pure synthetic polymer. This was basically used for intraoral wound healing applications. And this was with Sri Sathya Sai Hospital again in Bangalore. Next. The next one was for a researcher in MS Ramaya. So there basically he was utilizing this technology to develop beta TCP scaffolds. Basically these are bone-based scaffolds that he was developing. The application of the bone-based scaffold was to see how it can be used for treating minor and major fractures. We then went on to get a grant from BIRAC. So BIRAC provided us with a grant to basically bioprint human skin. So this is the human epidermis that we had bioprinted within our lab. The application for this is as an alternative to animal testing. It is as thin as the skin that you peel from your lip. Now this was basically, what the initial stages of our work, we were able to get to skin. Now what next? The next one was a partnership with Merck. So with Merck Pharmaceuticals in Germany, we basically built out a customized bioprinter, which allowed them to basically develop medium throughput bioprinting applications. So the medium throughput bioprinting applications, the first one that we looked at was just printing singular droplets inside a 96 well plate. All of you would have seen a 96 well plate. It's extremely tiny. The holes are extremely tiny. Imagine printing a single drop within that. So we were able to do that by developing a technology called anti-backlash technology. This allows you to mechanically control the movement of the machine, reduces the error rate that happens in the machine. The second one, they upped the ante a little bit this time. They said, okay, you have one drop. Why don't you print another drop right on top of that? This was for co-culture studies. Now, this was something that we were able to do in the second image that you see here. You can see a blue drop, and then you can see a red drop right on top of that. The main focus of that was to see how they could do co-culture studies, specifically tumor-oriented studies. The final one was where they wanted us to print lines right beside each other. They wanted us to print one line right beside each other, and this specific application was for, again co-culture, but they wanted to understand how healthy and tumor cells actually behave with each other. Next. So in the process, we garnered a few patents. We have a process patent for the bioprinting process of bioprinting of human tissues. We have 2 technology patents that allow for precise temperature control as well as for precise movement within the machine. But we also have a proprietary biomaterial that we have within the company. So the next one that I want to talk about is, over the period of the next few years, we garnered a few collaborations. We also had a few customers in terms of Central Leather Research Institute as well as JIS University. But I'm also going to talk a little bit about our research with Microsoft. Next. So why does bioprinting need AI? So the work that we're doing in Microsoft is mainly revolving around AI. So we're looking at how artificial intelligence can be used to image scaffolds in real time to understand, certain, discrepancies that can happen within the scaffold. It can be under extrusion of the biomaterial, or it can be an over extrusion of the biomaterial. Now, for us to understand that, we need to give the machine a pair of eyes and a brain. And that's basically what we built with Microsoft. We built a complete architecture system that would be able to take a picture of a scaffold and create hotspots where the scaffold is actually not properly printed. Next slide. So here you can see that there are multiple examples here. So the ground truth is basically what it captures. This is what we are also showcasing in Termis USA this year. So you see here on the right hand side, there are multiple breakages in the scaffold, and all of them are clearly denoted. Now the intensity of the red or the blue determines at what level basically the scaffold is broken, at which layer the scaffold is broken. Now that's where this technology plays a very key role. The next one is again the accuracy rate that you're getting. Any AI architecture that you're building, you need to look at the accuracy rate that you're getting, and we are getting at about 98.3%. So if you just show a scaffold to the AI, it'll be able to understand whether how good is your scaffold fidelity. Any researcher can come up here and show you fancy images of scaffolds, but the reality is when you read them under a microscope, in most cases, the scaffold has some kind of discrepancies. So that is what we are trying to show utilizing AI. We are trying to give the machine a pair of eyes and a brain. And in the future, we are looking at how real time we can correct the print as the print is going. Why is this important? Because when you're looking at bioprinting applications for clinical use cases, that is where AI comes into play. You need to understand that if you are going to create something for doctors, for clinicians, you need to have complete control over, the kind of precision your technology has. And that's what we are trying to build into Trivima bioprinters as well. So the next one is, we also had a few collaborations that also happened along the way. One is with Manipal University, where we are working specifically on how stem cell research can be included or can be driven via bioprinting. And more recently, we also partnered with High Media Laboratories, where they are distributors for India, and now we are also exploring global opportunities with them. So this is one work that we did with CLRI, where basically we printed a lumen structure. This lumen structure was basically utilized for their studies to understand how endothelial cells can be placed within this structure. Next one. This is the structure that we did for one of the IITs. So it's a very complicated a cortical bone architecture scaffold that they made. So as you can appreciate, there's a lot of complex architectures that are going within the scaffold. This is a 10-layer scaffold that was printed, and we had to optimize the print. The optimization of this print alone took close to around, about 10 days for us to just optimize each and every portion of the print. Next. This is something that we did more recently. This was for DY Patil Kolhapur. I don't know why it's not playing. Okay, it's fine. Go to the next slide. Yeah, so this is with Dr. Falguni Pati's lab at IIT Hyderabad, where Dr. Falguni Pati is printing the cornea. So we're working, our bioprinters at his location, and he's utilizing his DECM BioWings to develop these dome-shaped structures that can be used for his research. Right now he's in animal trials, and soon he will also enter into the clinical trials phase. So these are all our partners. So we have a lot of collaborations that we have with the government, as well as with a lot of researchers around India. Two of the most important things, we have Dr. Biman Mandal from IIT Guwahati, who is also on our board of advisors. We have collaborated with Dr. Falguni Pati as a company from IIT Hyderabad. And we have three basic machines that go out. One is Trivima Basic, which has a dual extruder, so I'm not going to be too commercial over here. Next one. We have Trivima Advanced, which has 3 extruders. That basically is the machine that you see there. In case you want to go and understand more about the machine, what the machine is all about, you can go and take a look at it over there. Next one. Then we have Trivima Pro. This is what we are right now shipping globally. Not only in India, we're looking at also the Middle East, Europe, as well as the Americas, to see how machines like this can be used for more of a clinical use case. So we are looking at how AI can be integrated into these machines. Our Pro machines are the ones that are going to showcase the AI power that NBL brings to the table. Next. Yeah, so basically you can work with up to 6 extruders. You have different modes of technologies, inkjet and all of that. But we'll tell you more about it at our stall. But let me quickly skip to the last slide. Can you just skip? Skip. Yeah, so yeah, this is where all our printers are put up at right now. So we are located in multiple locations around India where they're being used for multiple research use cases, such as, bone-based bioprinting, skin-based bioprinting, alternative leather. There's a lot of applications that people are exploring. But the more important thing is, as more and more bioprinters come out, we understand that the research is going more clinical. For the research to go more clinical, we need precise engineering. Normal commercial bioprinters do not allow you to go to a clinical stage. If you develop something and you get it to the doctor, the clinician will reject it because the device printing it is not exactly accurate as per medical standards. If you need to get there, you need to build a machine that allows you to get there. And that is what we are doing right now. Next slide. So this is where we also want to take bioprinting. A recent 100 crore grant that we have signed with the Department of Defense, especially DRDO. We are also working with ISRO closely with the Gaganyaan mission. The specific focus of the Gaganyaan mission is to put man on the moon, but the basic focus is to see how tissues or even meat can be bioprinted in space such that it can be utilized by astronauts. So this is the focus with ISRO and us. With that, just the last one more slide before this. Yeah. All of this wouldn't have been possible without my amazing co-founders and team members, some of them who will be here. Anything at all, you can reach out to us. We are happy that you came down to this conference, and anything at all, we'll be right there. And thank you.
Thank you, Mr. Piyush, for sponsoring us. Also for introducing Make in India product 3 bioprinter. So we are proud that they are developing Make in India products. Thank you. So our next speaker. Our next speaker is Professor Suchi Verma from Department of Chemistry, Delhi University, and her area of research is biofuel, biodiesels, sustainable materials and developments. Please try to finish by 5 minutes only. Our lunch hour started. Those who are hungry, they can proceed, because we have to finish lunch by 1 p.m. sharp. After that, no lunch.
Well, yeah, thank you. Good afternoon, everyone. I'm Dr. Suchi Verma from Ramjas College, University of Delhi, Department of Chemistry. My topic is, basically, I try to put it here as a solid waste management and the upcoming biorefineries from a cow dung. So the disposable of effective utilization of cow dung and undesirable solid waste for both therapeutic use and production of sustainable biochemicals. Next, please. Yeah, so basically, why I choose cow dung is a story, because I was talking to few people from the Goshalas, and they were saying that can you help us in making this money from 5 rupees to 50 rupees. So that's why I choose cow dung as a biorefinery from the upcoming field. So let us go for the availability. In India, 70% population is living in rural region, where cow is the principal cattle. It generates 7 to 15 kg of cow dung per day. Total milk producing cows are 195 millions and non-milk producing cows are 30 millions. So 500 plus million tons of cow dung are being used, wasted every year or thrown on the street, that creates not only pollution but also release methane. It's a harmful health and is one of the major reasons for global warming. Next is employment. We have know that about 20.5 million people depend upon the livestock for their livelihood. We provide, it is that livestock provide livelihood to 2/3 of the rural communities. It provides 8.8% of the population in India. Next, thank you. Next, ma'am. So, unique feature of cow dung. Why I choose cow dung? Because bio-waste is easily available. I took cow dung as my biomass. So, bio-waste is easily available. Big problem of its disposable, specifically in villages. It's a discharge hazardous pollutants, solid liquid gases. In solid, you can see it's microbes such as bacteria, yeast and molds. Liquids, arsenic, dioxins and chlorophenols. These all are basically your carcinogenic animals. And gases, you know, methane. Next, and E. coli, Salmonella, Listerine and cholesterol, and one of the major bacteria which are producing from the cow dung. So these organisms can affect people when they come across contaminated with soil, and under certain conditions, they can be taken into the plant tissues, and these will always risk that can be get sick. That is why we can get sick from the cow manures. The major 4 pollutants and hazards are basically 4 main gases produces from hydrogen sulfide, methane, ammonia and carbon dioxide. Methane is asphyxiates at extremely high concentrated. Ammonia, hydrogen sulfide irritate the eyes and respiratory tract. High concentration, it can cause severe high and irritant and respiratory tract. Now next is headache, nausea, dizziness, which you can even feel when the pollution is up or pollutants are up. Severe respiratory disease, which we have seen in one of the major reasons of corona is also we can see that it can be more prominent nowadays because of the pollution. And besides, unmanaged livestock waste produce the largest greenhouse gases emission, which is around ±18%. So this is very important slide because this is my research area, and recently I got 1 CR from the Ministry of Animal Husbandry on further reactions and further innovations on these products. So the average composition, I told you that one of the Gaushala people, you know, they challenged me that please help us in coming up the rates of the cow dung. So I just go to the analysis of that, and I got a very good result in that because already it is published data that it has cellulose, semicellulose and lignin up to in a dry cow dung from 49 to 54 or 60%. So this is my catch, so I took cellulose. So, you can change the slide. Thank you. So basically that's my area and I'm working on it, and right now I can't expose those results. So the next point is that basically it is how it is a biorefinery. So we know the traditional uses of cow dung, plastering of wall, partitions of rural homes for providing insulations. Ash, nowadays ash is using as a cleansing agent and in a beauty product also. More smoke generated from the burnt cow dung as mosquito repellent, biodynamic manuring, making plant ponds and agarbatti. Now the upcoming biorefinery which we are working on, and my upcoming fields for innovation from the cow dung is basically, I am working on bioplastic, biotextile and alpha cellulose. So many students of polymers are here, so we are working on that. So I really definitely they have a catch here. Silicon, recently there are the slides which they are working, they are talking about your that battery parts and all that. So they can use, we can extract, we try to extract the silicon from there. So silicon, ceramic, silicon-based semiconductors, biogas and biooil are very known products, fermentations like ethanol. MCF, microbial products, bacterial enzyme, obviously know that because we have a curd, we know the usage of curd. Probiotics are there in that. Pulp, paper, cardboards, obviously, and activated carbon filler bricks Activated carbon, we know that it is used in the reverse water system for the purification of water. So my submission is, oh my God, biorefinery based on cow dung can be profitable and sustainable upcoming industry. Technology assessment of engineering technology using very practical routes of converting waste. Industrial solution of economic development. Antibacterial, antifungal, probiotic properties. We can develop an entrepreneurship programs in the rural area. Last slide is there, sir.
Okay, last slide, please.
So my slogan from this is from ruler to rulers, and thank you. Thanks a lot.
Thank you for a lot of idea about biofuels, and regarding cow dung also you told how to get energy. And thanks a lot again. Please. Now our next talk will be by Dr. Prashant Singh, who is the co-founder of ATCG India. So he will tell about 3D printer from SailLink, also about ATCG. I request Professor Prashant to limit the lecture maximum 5 minutes. Please.
Hello, good afternoon everyone. I hope I'm audible to everyone. So thanks everyone, and then we are thankful for the organizers to give us the opportunity. So now that we have very limited time, so I'm just going to take you through the exact about the SailLink portfolio. So everybody here is aware about the 3D bioprinting technology. So we don't need to go into the basics of that. So our focus today here is to tell you that what it takes to choose a right bioprinter for you. So if you see into the market, there are multiple ways in which you can 3D bioprint. There are extrusion based, there are bio-ink based, there are light based. So SailLink is the pioneer in the 3D bioprinting world, and they were the first one to launch the most compatible bio-ink. So we supply multiple range of bioprinters which start from extrusion based and light based. So in India and global, we have lots of installations. We have more than 900 publications. Can you just check? Excuse me. Slides are not coming. Okay. So can you just one more? Yeah.
Audio, video, 1 baje jayega hamare saath, theek hai?
Next. Yes, yes. So this is the complete range of bioprinters what we have here. So if you see, we have the extrusion-based bioprinters here, and then we have the light-based bioprinters. So this fulfills the needs of all of your, bioprinting requirements. Next. Yes, sir. Yes. So like we were focusing on how to choose a right 3D bioprinter. Now there is it's a very easy call. I mean, it depends on what exactly you are trying to bioprint, what are the bio-inks available with you, and what are the downstream applications you are looking for. If you are looking for constructing hard constructs, say for example cartilage, bone regeneration, these kinds of constructs if you want to prepare, so we strongly prefer to go ahead with the laser-based bioprinter. And then if you want to handle work with the normal, say for example tissue engineering, personalized medicine, regenerative medicine, meat alternatives, if you are looking for these kinds of 3D bioprinting applications, then we strongly recommend to have the extrusion based. So there are multiple suppliers of 3D bioprinters globally. So what differentiates SailLink from rest of the world these are the points. So we provide the platform which is open biomaterial platform. You can use the bio-ink from any of the suppliers. We provide the patented, patented sterile environment. So it's a HEPA filter assisted, it's a UV light is attached there, so your cells would be safe there. Next one. And again, very important for any kind of 3D bioprinting is the way you control the bioprinting environment, which starts from your temperature, it starts from the safety of the material what you have here. So if you see, we have the multiple bio-inks available with SailLink, and if you can see here, we already have the publications in multiple fields. So these are the universal bio-inks available with SailLink. If you are interested in any of these applications like cartilage, skin, bone, muscles, so we have publications with all of these things. Next. So next one. So next one. Yes. So there are multiple references what we can present here, but because of shortage of time, this is the one what I like to tell you, show you here. In India, we have multiple installations. Pendurum Technologies is the one who is working on cornea. There are other institutes who are working on liver, cartilage. So there are multiple researchers in the India who are working on multiple dimensions. So we, from ATCG, we are open. We have the demo unit here, and you have our contact details now. If you have any, if you want to organize any kind of, lectures, road shows, webinars, workshops regarding 3D bioprinting, please feel free to reach out to us. Thank you.
So he has the bioprinter. If you want to know, they can help how it works. And also here is another bioprinter. You can have a rapid look to know how it is working and how it is helpful for the tissue engineering, especially Komal, you can see your work. A lot of people, they are working in the area of tissue engineering. They can see the demo, it is there. So you have opportunity to see this. And our next talk will be by Professor Sudeep Dasgupta through online. He is professor in NIT Roorkee. His area of research is fabrication of scaffold for different types of tissue formation and regenerative medicine. Let us welcome. लाइन हो नहीं रहा है तो जल्दी करो लाइन फोन लगाओ सबका Those who are still did not register, please register for Dev-Prayag journey. Here is the counter. You have to pay rupees 500 and then you enjoy the beautiful scene of the meeting of the Alakananda river and Bhagirathi river and the birth of the Ganga. So visiting Dev-Prayag is itself it's very holy for us. And also some t-shirts are available here at the cost of rupees 500 with BioHill and our Swami Vivekananda picture. You can see here in the stall, there are some t-shirts. If you want, you can purchase at the cost of rupees 500 only.
Hello. Yeah, I have seen it, but I have to share the slides, right?
So what happened?
Sharing
Is
slides.
linked or not? Linked? Now I can...
Sharing slide option is not available right now.
On the screen.
Share slide.
At 1 o'clock.
By 1, so please move to the dining counter. Ma'am, to the dining area.
दो चार लोग रह जाएंगे, अभी चलेंगे 5-10 मिनट बाद। बहुत चला गया ना, बहुत लोग चला गया। हो गया लिंक, प्लीज जल्दी। ऑनलाइन पैसा दिया बाकी टिकट भी बुक किया था, सब कुछ प्रॉब्लम आ गया आने, बहुत नुकसान। लोगों को कुछ अलग से पार्टी वार्टी मिलेगा कि नहीं मिलेगा? ऐसा नहीं, सच बता रहा हूँ, ये लोग 7 लाख देगा बोला था, कुछ कारण में प्रॉब्लम है तो सुदीप दास। Professor Dasgupta, can you hear me? We cannot see him. Professor Dasgupta, now he is coming. Professor Dasgupta, can you hear us? Yeah. उनका क्या म्यूट है क्या? बैठेगा तो सुनेगा ना। प्रोफेसर दासगुप्ता प्लीज, कैन यू हियर मी?
Speakers. Now. It's not there.
Professor Dasgupta, can you hear us?
Yeah, I can hear you. It's a problem with sharing of the slides. Data, where is it?
Or if you cannot share, you can speak. Huh?
Yeah,
we can hear you,
share.
but now we cannot see you. You are on the other side, not sitting on your chair.
I'm sitting on my chair.
I don't see you. I can see your vacant chair only. Yeah. Yes, now we can see you. Okay, let us welcome Professor Sudeep Dasgupta from NIT Roorkee. He is expert in nanoscience, nanotechnology, tissue engineering, regenerative medicines. And let us welcome Professor Sudeep Dasgupta with a round of applause. So, Professor Dasgupta, I request you to wind up in 5 minutes, please.
Okay.
Okay, thank you.
It's not visible, right? My slides are not visible.
You are visible. You can give some general talk also without slide. Professor Dasgupta.
Yeah, we are doing.
I think
just in a minute.
if some technical problem, you just tell about yourself, your research area, your achievements. And we will be grateful to listen that.
Just give us 1 minute. 1 minute, I am trying.
Yeah, 1 minute also a lot now for us.
Okay.
So you will get 4 minutes. 1 minute for this and 4 minutes for presentation. যাচ্ছ দেবপ্রয়াগ যাচ্ছ তো রেজিস্টার করে দিচ্ছ
Sliding desktop. Sharing window there is share. Windows there is share.
Professor Dasgupta.
Yeah, is it visible now?
Okay, now it is coming, it is appearing. Yes, we can see. So, please. Yeah, thank you. Please start.
How much time I have?
Now 4 minutes. 1 minute you have lost.
Okay, sorry.
Yeah, please go on.
It's a nice experience to share my slides and share my research experience with you. It's a pleasure to join all these colleagues and renowned professionals in my field, as well as educated audience to see in this platform. And I'd like to share my research experience. And of course, this is related to bioceramics and is composite for hard tissue repair and regeneration. This is the brief outline of my talk. As I don't have much time, so I'm just going into the introduction slides. So we are now considering biodegradable materials for bone defects. Biodegradable materials are so much useful because you don't require any secondary surgery. Also, these are bioactive. So, in terms of biodegradable materials, we have biodegradable polymers, biodegradable ceramics, and bioactive glasses, as well as biodegradable metals. So, my main focus is with biodegradable polymers and ceramic-based composite ceramics, mainly calcium orthophosphate family, like hydroxyapatite, beta TCP, calcium monazite, dicalcium phosphate anhydride, as well as calcium sulfate and other bioactive ceramics like bioactive glass. And in terms of biodegradable polymers, so we have natural biodegradable polymer or synthetic biodegradable polymers. In terms of natural biodegradable polymers, we have collagen, chitosan, gelatin, all of which have been explored by our students, as well as recent PhD work has been on silk fibroin and bioactive glass. And another PhD student is performing her research experiment on polyanhydride. It's a synthetic polymer. We have synthesized polyanhydride in our lab, and because it's a biodegradable polymer, and it can be impregnated with hydroxyapatite collagen nanoparticles, and this is being injectable. This is another advantage, that this is being injectable. This can fill up all the voids and defect sites. Before going into the further discussion about the materials aspect, we need to understand biomaterial cell interaction. And what is this? This is basically when a biomaterial is inserted inside a body, within a second there will be protein absorption before blood clotting, and then there will be protein absorption. And these protein absorption will allow cell to be attracted towards the biomaterial surface. Of course, the bacterial cells will be tackled by the immune cells, and the cell will see a conducive environment in the biomaterial surface. They will find a suitable environment, and then they will regenerate extracellular matrix within hours. Like in case of osteoblast or bone tissue engineering, there will be fibronectin network, growth factors, and this is the phase 3. And in the phase 4, there will be migration and proliferation and differentiation of the stem cells or osteoprogenitor cells to regenerate the tissue. So, regarding biomaterial and biomaterial or bone cell and stem cell interactions to regenerate new bone tissue, how stem cell regenerates? First of all, the stem cell sees the biomaterial and it adheres on the biomaterial surface, and then it proliferates stem cell. It gives according to the environment that is around its vicinity. It sees whether it will proliferate into it will differentiate into the progenitor cells like osteoblast progenitor cell, osteocyte progenitor cells, or osteoprecursor cell, and these will be consecutively differentiated into osteoblast and osteocytes, which are the matured bone cells. So, in the next slide, I want to show you some of the experiments that we have, some of the products we have developed in our lab, and this is one of the most important of those are this injectable paste for heart tissue regeneration. So we have taken methacarylated alginate and surface modified hydroxide nanoparticles. Surface modified, why? Because we need to get an interaction with the hydroxyapatite and polymer matrix, because otherwise hydroxyapatite will be agglomerated and there will not be uniform dispersion of the hydroxyapatite nanoparticles. So once we have got methacarylated alginate, of course silicon doping can be done so that osteomyelitis can occur at a rapid rate, because silicon has a positive role in upregulating osteoblast-related genes. And these are mixed with DMA media and is injected into the defect site, where after being exposed to the UV light or visible light of different wavelength, this is cross-linked, and then they have some mechanical stability so that they cannot be washed away. They cross-link, they form a very good interface with the old bone, and then the new bone formation occurs by the resorption of these injectable paste. Because all these bioactive materials, they form a very immaculate bonding with the old bone and new bone, and this bonding will allow the bone cell to grow inside and in, and then gradually resorb the material. So it is.
Dr. Dasgupta, excuse me, please finish up in 1 minute, please.
Okay. Okay, so this is alginate nanohydroxyapatite paste. That's what we have prepared in our lab. And then this is the polyanhydride nanohydroxyapatite paste, photocurable paste. We have used sevacic acid dimethacrylate and methacrylate 1,6-bis paracarboxyphenoxyhexane (MCPH), and this is supplemented with PEG-PEGDA. And the light initiator has been used as a camphorquinone or ethyl 4-benzoate, EDMAC diethylaminobenzoate, or chemical initiator as a benzoyl peroxide or dimethyl toluidine. Then these are the products that we have developed in our lab, and followed by this in vivo trial. We have used rabbit model for the in vivo trial of our product, and this is the radiology study of the material, and you see with the bioactive glass, the radio density is gradually decreasing and with increasing time of implantation, and bioactive glass shows the best promising properties in regenerating the bone tissue. Also with the histology, this is what more cellular activity and osteoclast activity are seen in bioactive glass gelatin chitosan implant after 3 months. Angiogenesis is visible in all the implant are more pronounced and then control after 3 months of study. This is a fluorochrome leveling study, and this is fluorochrome leveling indicates the new bone formation, and you can see more new bone formation occurs in the composite supplemented with bioactive glass. And this is the electrospan fibers using chitosan, gelatin, PEO, and monatomic nanoparticles that we have prepared, and is the effect of viscosity in getting the uniform fibers, electrospan fibers, in the chitosan gelatin monitoring scaffold using electrospinning technique that we have prepared. And this is the osteocalcin expression study of gelatin and dicalcium phosphate nanofibers, and which shows that at 7 or 8% DCPA content, it expresses more osteogenic capacity more differentiation capacity by eliciting more osteocalcin expression and more osteogenetic properties. These are the conclusions. So, photocurable alginate surface modified oxidative MPs, waste-based and injectable phase exhibit a promising potential for healing complex shape bone defects and disorder, methactylated monomers of sebacic acid and 1,6 carboxymonoxide were used for photovoltaic polymer paste. Both silicon and transition to hydroxyalcohol nanoparticle incorporation and RHBMP2 was loaded in the polymer paste, which showed significant improvement in oxygen capacity in vitro as well as in vivo. Yes, thank you. If you have any question, I'm ready to accept and I will reply whatever way I can.
So, thank you, Professor Dasgupta, and thanks a lot for presenting your wonderful research work. And maybe next time we meet face to face, and then we'll have more discussion. Thanks again.
Thank you. Thank you.
So, I request all of you to proceed for lunch. It will be ended at 1 o'clock sharp. And after that, we will proceed for day prac. Those who has not yet registered, please register at the desk. The registration person is there. Thank you. So we end up this session now. Again, we will meet 8 o'clock here for poster session, for our cultural function, and also award ceremony. And okay, thank you.